Welcome. Welcome. Good afternoon. Good afternoon. We just want to give it a straight start. My name is Matthias Leopold. I'm currently the head of School for Agriculture and Environment. And before we give the official start, I just have a little bit of housekeeping to do. Just be reminded, in the unlikely situation of an emergency, please follow the exit doors here to your right, but also to the left and in the back. And in case you would need some toilets, please, that would be outside of the foyer into the other building. There are toilets to the left and the right if you continue there. Welcome, everyone. Good afternoon. Before we start with our official program, I would like to follow the customs here at the university and acknowledge the land and country where we stand today. We acknowledge UWA is situated on Noongar land and that Noongar people remain custodians of their land and continue to practice their values, languages, beliefs, and knowledge. We pay our respects to the traditional owners of the lands on which we live and work across Western Australia and Australia. Along with tens and thousands of years of Noongar people's stewardship to the country in the past and for the future generations, I also want to acknowledge many people in this room who have shared some spaces and showed our intent together to sit on country with the vision of care and commitment to the land. It's really with great delight and pleasure to welcome everyone who joined us in celebrating 50 years of education, training, scientific vision, and service of Emerita Professor Lynette Abbott, who likes to be called Lynn. I welcome Lynn's family, her husband, Dr. Ian Abbott, her brother, Peter Jones, their family friends, Nicole Robson, Lynn's previous PhD students, Professor Ann Nurbaiti and Professor Inan Junna, who arrived from Indonesia for this event. Emerita Professor Cheryl Elizabeth Prager, Emeritus Professor David Lindsay, Emeritus Professor Hans Lambers, Emeritus Professor Stephen Powles, Professor Simon Cook, Dr. Graham Robertson, Ben Biddup, Dr. Mark Sweetingham. I also like to welcome our colleagues and students from UWA, from CSIRO, DPIRD, Eddie Cowan University, Curtin University, Murdoch University, and industry partners. Now I would like to invite the University of Western Australia's Senior Deputy Vice-Chancellor, Professor Tim Colmer, to formally open the Grounded in Soil Science 50 Years of Emerita Professor Lynn Abbott in Soil Science Symposium. Tim, the floor is yours.
Thank you, Matthias. Welcome, everyone. I too acknowledge the Whadjuk Noongar people as the traditional owners of these lands and pay my respects to their elders past and present. It gives me great pleasure to formally celebrate the remarkable milestone of 50 years of service to education, research, and indeed dedication by Lynn to UWA. Lynn did her PhD at Monash University and joined UWA as a research fellow in 1974, and since then made WA her home and UWA the centre of her passion for education, scientific discovery and community service, particularly in the subject of soil biology. Over these years, Lynn has been recognised as a global leader in soil biology and has earned many accolades, nationally and internationally, including the inaugural General Jeffrey Soil Health Award, a Fellowship of Soil Science Australia, and as a Fellow of the Australian Institute of Agricultural Science and Technology, and various UWA teaching awards as well, including most recently in 2022, an award for research mentorship, demonstrating her continued strong and valuable contributions to the university. Lynn's impressive tenure at UWA extends from being in the Soil Science and Plant Nutrition Department to then as Winthrop Professor in the School of Earth and Environment, and now as Emerita Professor in the UWA School of Agriculture and Environment. And during this time, Lynn has served our university in various leadership roles, including as Deputy Director of the Institute of Agriculture, Vice and Deputy Dean of the Faculty of Natural and Agricultural Sciences, and as a member of many working committees in the Faculty of Science and various university boards, such as Academic Board. So indeed, as a young academic, I enjoyed the experience of joint research supervision with Lynn, and I also would like to acknowledge the important leadership outcomes from Lynn's faculty roles here at UWA. Lynn was mentored by the late Alan Robson, and Lynn has also made tremendous contributions to improving the working culture here at UWA, and is among the pioneers of some changes in culture at the university. Lynn was the first chair of the planning group of the UWA Leadership Development for Women program, playing a fundamental role in its establishment and staying involved for 20 years. Lynn was also instrumental in securing the grant to establish Unicare. And Lynn worked closely with the university architect on the building plans for Unicare. As a passionate scientist, science communicator and mentor for the next generation, Lynn established a mentoring program for agricultural science students in collaboration with the Ag Institute Australia, which matched students to industry mentors. And that was a precursor of our current Uni Mentor program. Lynn's contributions are recognised nationally. She is one of the 6 women scientists celebrated at the Questacon Museum in Canberra. Lynn is also deeply connected with the farming community across Australia, including local landcare bodies, and she continues to conduct regular workshops with farmers and advisors across Australia, and served as a member of the Australian Government Domestic Offsets Integrity Committee. Lynn is also chair of the WA Crawford Fund Committee. It's based on her profound impact on soil science, her dedication to mentoring, and her unwavering advocacy for diversity and inclusion in science that it's my pleasure to let you know that the UWA Senate has approved to award to Lynn a Chancellor's Medal. Lynn will receive the Chancellor's Medal at the 13th of December 2024 graduation ceremony. So Lynn, I thank you for all that you have achieved and all that you have done to help others during your remarkable 50 years here at UWA. We look forward to your continued contributions. So please do enjoy today the well-deserved recognition and the celebration. The program of speakers looks great. I hope that you will all enjoy this event and join me in congratulating and thanking Lynn.
I'd like to give a couple of testimonials as head of School of Agriculture and Environment. Emerita Professor Lynette Abbott's 50-year career has been a remarkable journey as a passionate scientist, science communicator, and mentor to the next generation in the fields of soil science and soil biology. Renowned globally, her pioneering research on fungi-plant interactions and soil management has significantly advanced the understanding of soil health and sustainability in disturbed land environments, an area largely overlooked by mainstream enterprises at the time. Her exceptional contributions to understanding plant-soil relationships through both fundamental and applied research, as well as her efforts in disseminating this knowledge to students, farmers, research managers, have been widely recognized. The achievements were recently honored, as we heard, with the UWA Chancellor's Medal, as announced by Tim. At the faculty level, Professor Abbott has held several leadership roles, including Head of Soil Science and Plant Nutrition from 1992 to 94, Head of School of Earth and Geographical and Environmental Science 2004 to 7, and then again 2009, Associate Dean and Deputy Dean 1998, 2002, and Vice Dean of the Faculty of Natural and Agricultural Sciences from 2010 to 2012. She also served as the Deputy Director of the UWA Institute of Agriculture and as coordinator of the Rio Tinto Masterclass program. Additionally, she was a visiting professor at Langshu University from 2016 to 2023 and the University of Putra in Malaysia from 2019 to 2021. So she traveled and is still traveling a lot, which is great. Currently, she serves as a WA State Committee Coordinator of the Crawford Fund and regularly organizes networking and career events for students. This is where a lot of the passion of Lynn still goes out to the next generation. As a mentor and educator, Lynn has profoundly influenced countless students and colleagues. She has supervised over 30 PhD students, published more than 170 research papers with over 10,000 citations, and holds an age index of 58 as given by the Web of Science. Known for her generosity in sharing expertise and her ability to inspire others to reach their full potential, her extraordinary contributions to mentoring and education have been recognized with numerous awards, including the Carrick Citation for Contribution to Teaching and Learning and the Excellence in Teaching Award. Professor Abbott has also played a significant role in various national and international committees for awards and prizes in soil science. Her contributions include serving on the International Mycorrhiza Society Awards panel, the Soil Science Society of Australia, and the Ag Institute Australia as an assessor for the National Student Award. She has also chaired and been a member of the International Union of Soil Science Division and Standing Committee on Prizes and Awards. However, her work extends far beyond academia, impacting society and in a deeply practical and personal level. Professor Lynn Abbott actively participates in numerous community and agricultural initiatives, including city farm events, National Science Week activities, Kids to Country programs, perennial pastures establishment and regenerative farming workshops, and the Institute of Agriculture's Future Farm 2050 Project Committee. She is also a dedicated volunteer to the Royal Agricultural Society of Western Australia, assisting as a scorer in annual school farm skills competitions. This list is far from exhaustive. In the agricultural sector, Professor Abbott has fostered strong connections with farming communities across Australia, including local land care groups. She mentors aspiring soil enthusiasts, providing guidance and opportunities to deepen their knowledge and practices in soil health and sustainability. Passionate about translating scientific theory into practical applications, she regularly conducts workshops with farmers and advisors nationwide. Recently, she developed a soil health app, a digital tool designed to simplify access to complex soil health information. Just last year, she conducted 6 farmer workshops in the northern and southern wheat belt of Western Australia and regional South Australia. This is merely the tip of the iceberg. Everyone who has had the privilege, me included, the privilege of working with her knows the immense scope of her tireless advocacy for soil science and soil health in agriculture and beyond. It is why we are here today, to celebrate her extraordinary 50-year contribution to the field. Now, I would like to invite Don Burnside from the Ag Institute Australia WA Division Committee to share a few words on Lynn's contributions.
Thank you, Matthias. My name is Don Burnside, and I'm a member of the Ag Institute of Australia, which was formerly known as the Australian Institute of Agricultural Science and Technology. And it's a great honour to be here today and to reflect upon and celebrate and congratulate Lynn on her 45 years of service to the Institute. And Lynn, it's clear you have a lot of friends here. Lynn joined the Institute in 1979, and as far as I know, she was very shortly afterwards invited to join the West Australian Division. She has been a member of the West Australian Division ever since, and I expect you to continue being a member of the West Australian Division, Lynn. She served as the chair of the WA Division in 1990 to 1991 and in 2014 to 2015. These days we tend to run the WA Division as a collective, and so we share the responsibility for chairing the committee and organising events. She was a member of the Awards Committee for the WA Division in 2017-2018, 2018-2019 and 2019-2020. And she's continued in an informal role in supporting nominations for fellows and awardees of excellence throughout that period and until the present. Throughout her time as a member of the Institute and as a member of the WA Division Committee, she has had a very strong focus on encouraging student membership and participation in the Institute, in particular reaching out to student bodies at UWA, Murdoch and Curtin. She has been very active in helping organise the Young Professionals in Agriculture Forum, which is held annually, which is a red letter event for us, in selecting leading students to compete for national awards. And I note I've seen at least one of our past award winners in here today, and I'm sure there are others who have been participants. She was made a Fellow of the Institute in 1993, and together with Dr. James Fisher, she has coordinated the very successful Careers Night for students, which we hold annually in April of every year. And this commenced as a fairly modest affair back in the early 1990s. And now it is a major activity for the division, with about 80 or somewhere between 60 and 80 students being able to attend a major workshop type event at the Royal Agricultural Society, where they can do speed dating talks with up to 15 employers and universities. And it's a hugely popular event, both with the employers who come back every year and also generously fund the event, and also with the students. As well as organising this with James Fisher, Lynn has also dispatched 3/4 of the way through the event to go and get the pizzas and the refreshments, and she comes back and the pizzas disappear in about 0.5 of a nanosecond. Other activities that Lynn has been involved in are the Discover Dirt exhibit at the Perth Royal Show in 2015, and in organising field visits for Institute members and sundowners for fellows and other events where members can get together and learn something about what is happening in agriculture. Lynn also links our division with the Crawford Foundation, as Matthias mentioned, and we have had joint functions associated with conferences at which students supported by the Crawford Foundation have spoken. And I, for one, have found those enormously interesting. In summary, Lynn's length of service and her ongoing contribution are praiseworthy, and on behalf of the WA Division and its committee, we congratulate you, Lynn, and thank you sincerely for your years of service and for those still to come.
Thank you, Don. The Australian Society of Soil Science Incorporated, also known as Soil Science Australia, is a not-for-profit professional association dedicated to soil scientists and the responsible management of Australia's soil resources. Lynn has been a long-standing member of the organisation, particularly the active WA branch, which promotes soil science awareness throughout the state. Please join me in welcoming George Mercer, President of the Soil Science Australia WA branch.
Hi everyone, just a few brief words from me. I've been granted the honour of sharing Lynn's extensive and passionate commitment to Soil Science Australia, but I feel like a bit of an imposter in this room amongst the great wealth of connections that Lynn has made throughout her career. You see, Lynn was the Federal Vice President of Soil Science Australia in 1999 and 2000, and then the Federal President in 2001 and 2002, a national champion and protector of soils at about the same time that I was still eating it. So I won't try and speak to a time from which I am not. But I can tell you that Soil Science Australia is a non-for-profit society that is built and operated on the volunteered time that is donated by busy professionals towards soil stewardship. It's been 22 years since Lynn was at the helm of Soil Science Australia, and guess what? In her Emerita role, she still actively contributes to the local monthly committee meetings, planning sessions, AGMs, and community lectures. So what does that tell you? I think I'm yet to attend an interstate or international conference where Lynn's influence is not felt. Like the legacy of soil organic matter left behind from the microbial activities of the past, Lynn's wisdom and energy has absorbed and aggregated into a diversity of stabilised soil knowledge. It is for this eminent service to soil science that we're delighted to award Lynette Abbott with honorary life membership on behalf of Soil Science Australia. Congratulations, Lynn, and thank you for your service.
Thanks a lot, George. As a passionate advocate for women in science, Lynn has been an active member of Graduate Women WA, a century-old organisation dedicated to empowering women and girls through lifelong education. Graduate Women WA provides competitive scholarships for women across diverse fields of study. Please welcome Graduate Women WA President Felicity Farrelly to share a few words.
Thank you very much. I'm Felicity Farrelly. I'm actually also an agricultural scientist, but I went a different direction than in the labs. But wonderful to see a few of people I recognise from 40 years ago. Lovely. Today, we're not only honouring an agricultural scientist, but in our context in Graduate Women Western Australia, we regard Lynn as a woman of wisdom in education, excuse me, one who is an outstanding mentor to the Graduate Women WA Association. Lynn Abbott has been a member of Graduate Women WA for over 40 years, providing wise counsel, strong representation for women, and clear leadership. Lynn joined what was then the Australian Federation of University Women. Some of you might remember they were the ladies who hired out the gowns when we used to graduate. And they all caught us. I think we all became a member if we were a woman for at least a year. Well, Lynn came across a friend who was Dr. Ines Tomarup, whose mother had been an early Queensland member of the association, and Lynn joined then. Personal connections continue to be the way for women to make contact within our organisation, and that's how we get a lot of our members. Look, if I digress for a moment, and Lynn did say I could talk a tiny bit about graduate women, it's the importance of, particularly in the early years, and also the continued relevance to this day. The International Federation of University Women was established in 1919. The first group got together in Australia, in Sydney, in 1922. And in 1923, the West Australian Association of University Women was established. So we're actually in our 101st year. We actually have passed that. And the first president for that was Dr. Roberta Joel. Dr. Joel was the first woman to establish a medical practice in Perth, and she also was a close associate of Edith Cowan. Lynn has encouraged the current members of Graduate Women WA to continue the legacy of our founders. Lynn continues on the tradition of keeping our membership relevant. She successfully encourages young women to join Graduate Women WA and then brings along others in a collegiate and supportive way. Lynn has been extremely involved in supporting graduate women right from the time since she became a member in that way. Lynn has held the position, particularly important to the Graduate Women WA, as the Graduate Women WA Education Trust, and Lynn has held that position since 2013. In probably a decade before, she was on the board, but in 2013, she took chairmanship of that. The funds held in the trust enable us to provide scholarships and bursaries to women who are completing their PhD research. In 2024, we've just given out 6 scholarship bursaries to the total value of $30,000. The trust also handles a behest from Leah Jane Cohen, which enables Graduate Women WA to provide a scholarship of $5,000 to assist a female law graduate, and that is given to complete their legal training. Lynn and the other trustees must ensure all legal and tax requirements are met in the awarding of scholarships bursary each year, that we continue to follow any new requirements that are placed on us, having a trust, and Lynn has provided strong and calm counsel during some years where the ups and downs in financial markets have presented challenges. Our trust is in a very strong position now, and we will be able to continue providing scholarships bursaries way into the future. Separate from the trust bursary scholarships, we also do provide for $6,000 for people, Department of Communities, to support young women, and we also support 2 young girls as part of the Smith's Family Education Support Program. And this has come about by discussions within our committee, including Lynn, where we want to ensure that we are relevant to others, not just in the tertiary environment. The tertiary awards are given out to all opportunities given out to all universities. Did start at UWA, but in the last 10 to 15 years, women from all universities have applied. We had 86 applications this year. Lynn, and I'm going to talk now about you, Lynn, in a closing manner. Lynn mentors the community she lives in, and I've been listening to all of you, and she is a fantastic mentor. Whether a student, staff member, or those of us at Graduate Women WA, her expertise and knowledge are widely sought, not just in agriculture, but in university function. She's been an administrator, and she's out there on the farms, helping people there too. She also came into the school system with a little experimental thing on how to identify soil animals. I was teaching one stage to year 9s who were actually captivated by these little tiny creatures that came out. So she's actually got out there into the secondary education and tried to instill in them an awareness of the importance of our particular things of our soil. She has also brought in a number of wonderful new members recently into our Graduate Women community, and only last month, I'm very happy, I've seen Hira here today. Hira Shekot was elected as our new Vice President. So we're looking forward to Hira running our association in the near future. Today, excuse me, today Graduate woman honours Lynn. She has been a wonderful member and our friend, a passionate soil scientist, we know, but she has achieved great outcomes, not only in agriculture, but also for women in education. And we particularly give you our grateful and special thanks. But before I close, I am reminded that we should also put forward to the people that Lynn also volunteers to do some, I think it's knitting, and heads up to 2J twice every fortnight, volunteers in a shop there, and the money raised goes to sending jumpers to India. So Lynn, I don't know where you get the time to do all this, but congratulations. Thank you.
Thanks a lot, Felicity. With that, we conclude our testimonials, and I hand over to Hira, and she will guide us through the following program.
Thank you everyone for joining us today in Grounded in Soil Science, celebrating 50 years of Emerita Professor Lynette Abbott's service and research. Now our program will follow the pattern of a soil symposium, where we will discuss about the advances in soil science in the last 50 years. Our first speaker is Manager Soil Science and Crop Nutrition, Department of Primary Industries and Regional Development, Chris Gazey. Chris focuses on achieving farm productivity and profitability by overcoming soil constraints. He has 40 years of experience in agricultural research in WA, starting from the University of Western Australia in soil microbiology, followed by 30 years of DPIRD service. Chris has been instrumental in facilitating improved farming practices, particularly using lime to treat soil acidity. Chris also holds the position of co-director of Soil West, which is an alliance between Murdoch University and DPIRD with other universities and CSIRO. Please welcome Chris Gazey.
Good afternoon. Well, it's not every day you get to come and celebrate someone's commitment to a service like we are for Lynn today, so thanks for that. And look, the really important thing here is that it's a privilege for me to be able to talk about where our soils work has gone since I started. And those first 11 years where I was working with Lynette Abbott and Alan Robson are really important to me, and I think that's something that I've taken with me through the rest of my career. So we go back to 1983 when I started working with Lynn, and I've actually returned that slide today to Lynn. That's how we used to do PowerPoint, the 35mm negatives. So there was no getting it done the last night before, you had to be well organised. So what I take from the time that I worked with Lynn here at UWA over those 11 years was a collaborative nature, and we've already heard about that, about how we cross-discipline research. And that's, I think that's been really important, and it's the way I've taken sort of my approach to my career and the time spent and the people working in soils that I've had to deal with. The other thing is attention to detail. So the importance of making sure that we're doing the science carefully and properly and accurately. And I think that is essential at whatever level you're approaching the research to maintain that integrity. Personally, I look back on those years through the formative years of my early career and see the mycorrhiza lab. We had 17 people at times. We had international postgraduate students. I see Sato in the audience and Chris. There was such a vibrant and active lab looking at mine site rehabilitation, pastures, plant nutrition. And when you're starting a career, you don't actually realise that that is the way, that's normal. That's just how big that is. You think it's just normal. That instilled to me, a lot to take forward. So I'm going to go back now and try and work through a timeline from the 1980s, when the majority of the grain belt, I guess as we know it today, was 60%. We're not going forward, are we? Because I'm not quick enough. There we are. Right. in 1980, 60% of the grain belt was pastures, and most of it was under a wheat lupin or a wheat pastures rotation. And the yield was around about a tonne of wheat per hectare. As we jump forward, say, 30 odd years, we've now got an area to 2010. We have an area where the land is dominated by cropping, 60% cropping, and some of that is 100% cropping. You know, some farmers have no animals on their thing and on their stuff, and we've increased those yields to 2 tonnes. So here's a chart of wheat yields that I've taken from the Australian Bureau of Statistics. A massive jump through the 1980s in terms of yield as we moved to more cropping and more inputs, and then another increase sort of after about that 2010 times in terms of yield, which now averages out at around about 2 tonnes to the hectare of wheat. So that's a big input, big increase, and a big ask from the soils that are being cropped. Of course, all of that input is not due to just soil management. You know, we have increased or improved genetics. We've got increased agronomic factors, early sowing, no-till. We've had a much better crop protection against weeds, pests and diseases. And all of those things combined to achieve the potential that is available. But really it's come at a cost, and I think, as we know with everything, if we stick to something, it's going to generate its own problems. So the strong no-till system meant that there was no mixing of soil nutrients and mixing in of lime that had been applied. We required more lime because of the increased production and the increased use of fertilisers, ammonium-based fertilisers in cropping, led to soil acidification, and that picture on the side just shows how you can get some intense acidification in the subsurface layers. That's been a big area that I worked on. But with this increased yield, you also had increased size of machinery and therefore soil compaction, and that's got progressively deeper as well. If we haven't had things like controlled traffic, which has just recently become a lot more practical. Also with that lack of soil disturbance, as we've been in a no-till system, we've had increased development of water repellents on the surface soil, reducing the amount of water that could easily access and run in, and also in some cases a rundown of nutrients. In particular, one that's being looked at now is potassium. So some of those with the extra, obviously, removal. A couple of constraints that we're addressing now that are not so much associated with the increase in production but exist on different soil types are acidicity and transient salinity. Through the 2000s up to 2018, most of our work was, we had big collaborative projects with UWA, with hostile subsoils, with Professor Bob Jilks. We had Soil Constraints West, a lot of interaction with Murdoch University there in terms of having PhD students. And the work was characterised by looking at individual constraints, but focusing on their understanding of their understanding the fundamentals of how they occur and what could be done about them, but also practical research on individual constraints and how could we correct them more efficiently in the field. And obviously, if there's just one constraint present, it's relatively easy to fix. You know, add lime to fix acidity as long as you can get it in the right place. And those sort of things happen. But as we continued that work, we weren't getting the responses that we were expecting, and it became obvious that many of these soil types that we have across the grain belt of WA have multiple constraints. And the image there on the screen shows that we've got water repellents in the surface, we've got subsurface acidity, so low pH below 10 centimetres, and we've also got high levels of compaction. And unless you can deal with all of those constraints in at a single time, you're not going to get the benefits of treating any one or a couple of them. So by way of an example about just where do these constraints occur, this is some important work that we did with industry, with Precision Soil Tech, with funding from the federal government mainly, from 2005 through to 2012. And the important thing here is that these risk maps, or these actual maps of understanding where low soil pH is occurring, are made up from real data. We looked at 94,000 topsoil samples and 67,000 topsoil subsurface samples from 10 to 20 and 20 to 30 centimetres, and that was from over 93,000 sites. So that level of data, that level of currency, really gave us a lot of confidence in what we were looking at in terms of the distribution and how this constraint was impacting agriculture. The darker it is, the worse it is the theme there. Two more maps of other constraints, and these are taken from the detailed soil polygon database that Department of Ag DPIRD manages, show us that where we look at the soil types and we understand the risks of which soils are prone to compaction, that we can see many of the sandy soils are highly prone to compaction in the dark purple, and also which ones are susceptible to water repellents also in the dark purple. What this map shows, if you think back to the soil pH map, is that there's a lot of interaction, a lot of co-occurrence of these constraints, and that's what we need to be able to deal with. So we termed an expression called strategic tillage. After you've had growers strongly committed to no-till for 25 to 30 years, suggesting that we bring out a moldboard plough and turn it all over was not the most popular thing. So we needed information to back this up, and through a whole series of trials looking at strategic tillage, making sure that we could assess what constraints were there and then address those constraints in a single once-off operation, and then of course go back to a no-till and a no-till farming situation for all the benefits that entails, including the controlled traffic, to preserve that loosening, to preserve that mixing and everything that we had created. Soil amendments that we used, you can see them there, but typically, clay, lime, organic matter if it's available, gypsum on the more sodic soils, and also in some cases the distribution of nutrients, redistribution of lime that would have been applied over the years but had not made its way through the soil profile. All of these gave massive benefits to us. If we look at the adoption that's happened over that time, this first set of the darker bars are from 2019, at the beginning of a new project that we had, looking at what was the adoption of amelioration practices. About 50% had looked at had adopted mechanical tillage, and about another quarter had non-mechanical tillage applying amendments. The biggest jump by the end of that project was that the mechanical tillage had increased up to about 2/3. And if we delve into that a little bit further, we can see that a lot of that was due to an increase in deep ripping. The jump there was actually not only just deep ripping, but deeper deep ripping. So rather than just down to the normal 30 centimetres or the typical 30 centimetres of depth that deep ripping tines would reach to remove compaction, we demonstrated that really, with the bigger machinery, that compaction was reaching down to 50 centimetres and needed addressing. And growers had the size of machinery and the equipment to be able to do that and to achieve those gains. What do the benefits of these practices and everything that have been adopted by growers translate into? And this is some work that in that same project we were able to do with Farmanco and with Plant Farm, looking at de-identified grower data and dividing it up into two sets, those that had adopted the soil management amelioration package and those that were still in what we'll call standard farming, that hadn't yet fully adopted some of the things that I've spoken about. What that meant over a 10-year period is that the average water use efficiency had almost got by 3 kilograms of wheat grain yield per millimetre of rainfall. Now, that may not sound a lot to you, but when we translate that into dollars per hectare of effective rainfall, we can see that the amelioration group are still well ahead. Obviously, in a 2019 year, you'll be looking at, if it's a poor year across the board and very little production, then everyone's going to be struggling. But in general and across the whole of that cohort over the 10 years, growers who adopted that package were $100 per hectare per year better off in terms of operating profit. That's just a huge result and a huge incentive for people to be assessing what they've got in terms of their soil types and the practices that they need to work with to get there. Obviously, when we've increased the potential to be able to grow those bigger crops, we need to be sure to make sure that they're fertilised to achieve those yields. And this work is showing that where we have a deep ripping treatment on the chart, we continue to increase yield and we continue to increase the net yield up to a point. However, where we've got an untreated or a constrained profile, that increase in nitrogen means maybe a little bit of an increase from nothing, but not much after that. And most importantly, what it means is that the extra money that you're spending on the fertiliser, you're not actually getting return from the extra yield that you're growing. It's just being spent on the extra fertiliser. And that's really, in parts, an extra risk to growers if those seasons don't deliver. So you really need to be very mindful of how much fertiliser is there and the opportunities and the potential. We've not just stopped at the strategic tillage point, which is the adoptability of the things that we know we can do, but we have looked at what could be achieved if we could do more. So if we could achieve a removal of all of the soil constraints down to a depth of 80 centimetres, albeit uneconomically, and you can see one of those examples given there in the top picture. Big backhoe diggers removing soil layer by layers, reconstituting it with addressing all of the constraints that we are facing in those soils, and indeed adding extra nutrients, adding extra lime where necessary. We can see in that top table that we go from a paddock control of around 2 tonnes to the hectare as an average from 9 cropping years up to 3.9, so almost doubling that yield where we remove all of those constraints. Now, we've probably opened a bit of a lollybag and we are getting a huge a boost. We're yet to see just how long that boost will go, but we get a similar picture as I've just painted for the wheat in the canola yields, a number of years as the example. But the picture at the bottom tells a story. Those 3 green strips are the what we call re-engineered strips that are still green, and still biomass and still grain being filled there against the strips next door to them, which are the unameliorated controls that have already SNESed the same as the paddock. And again, almost a doubling of yield. That work has opened people's eyes and said, well, there is still some more yield possible and more yield to be had. And we have engaged in a SWAN, Soil, Water and Nutrition collaboration with GRDC for the next 5 years. This work is going to be aimed at developing techniques and practices to take what we've learned, or take a lot of what we've learned from the re-engineering of the soil profile and convert that into adoptable packages so that we can achieve most of the aims that are available. Not quite sure how I'm going for timing, so I'll just watch that. Obviously, we also need to fine-tune the nutrition, as I've spoken about, and make sure that the fertility of the soil is correct. Another big component of that SWAN collaboration is things we've heard about already this morning, is the translation of that information through various courses, various education practices, through our engagement with Soils West and Murdoch University and all of the other universities and people, consultants. So this has been my final slide. I've got a comment on soil health. It means so many different things to so many different people that I think that's where sometimes we get confused. But really what we're trying to say is that all of the processes that I've talked about, all of the production that we've talked about is underpinned by soil biology functioning. And we need to make sure that that is happening, and it's possibly even more important once we've removed soil physical and chemical constraints and allowed the crops to reach some of their potentials, and maybe allowed that biology to also reach potentials as well. So I think with that, we also need to make sure that it's done in a sustainable way, making sure that we meet market expectations. And I'd just like to thank Lynn again for her contribution to my career and the things that it's made me look at in terms of how I deal with people, how I deal with and collaborate with different people. And thanks, Lynn.
We might not have time to take questions during the presentation, but you all are welcome to continue the discussion during the high tea. Our next speaker is Dr. Louise Barton. Dr. Louise Barton is a soil scientist and associate professor at UWA School of Agriculture and Environment. She has been researching in nitrogen cycling in diverse landscapes and climates. She brings the understanding of soil biological, chemical, and physical interactions for minimizing nitrogen emissions and leaching. Her current role heavily revolves around teaching and conducting field trials in collaboration with industry, Department of Agriculture, and Grain Research and Development Corporation. Please join me in welcoming Associate Professor Dr. Louise Barton.
Good afternoon, everyone. Oh, what a wonderful turnout and what a wonderful event. Lynn, I was thinking back, you know, I first met you 35 years ago as an undergraduate second-year soil science student, and I just can't believe that I've been given this wonderful opportunity to talk today about my research. So I'm going to be talking about some multi-year field-based research that we've done here in Western Australia. And I'd like to think that it's a good example of how understanding the soil biology of our soil is not just scientifically interesting, which is what most of us are motivated by, but it's also beneficial for our growers, understanding their soil biology, and the broader agricultural industry as well. So I'm going to be presenting a pack of data today, and I make no apologies for that, because I want to, appreciate not just the work that we've done here at UWA, but also acknowledge all the technical support that I've received over the years by the various members of my research team, acknowledge the various sources of funding that we've received to ensure that we've been able to continue on with that work. So today we're largely talking about research that's been done here in the West Australian grain belt, and I don't need to tell you all how extensive that land area is. As Chris has shown, extends from Geraldton in the north through to Esperance in the south. Here in the grain belt, we have a semi-arid Mediterranean-like environment. We experience hot, dry summers and mild, wet winters, which is when we grow our crops. We have highly weathered ancient soils. And as Chris has nicely explained, these soils often present us with challenges due to the constraints that they give to agricultural production. But despite that, this area can produce up to 40% of the national grain exports. And this is due to lots of various innovations, which I'm very grateful that Chris explained to us all. And one of those that Chris pointed out was due to increasing use or the introduction of fertilisers, and particularly the use of nitrogen fertilisers. Now, of course, when we apply nitrogen to our soils, the aim is that it will be mainly taken up by the crop, and some of it might be also incorporated into the soil organic matter and subsequently be released later for other crops. But there's always the risk that some of the nitrogen that we apply will be lost from the soil. And one of the loss pathways that I want to talk to you about today is the one that involves nitrous oxide. Soil nitrous oxide emissions result from a variety of soil biological processes that can occur in the soil. They're just soil microbes going about their everyday, day-to-day business. The two key processes that we talk about are nitrification and denitrification. Nitrification occurs under anaerobic soil conditions. By contrast, denitrification is facilitated by soil microbes when the soil becomes anaerobic, for example, after, say, heavy rainfall events. And then there's other processes like nitrifier, denitrification that can also contribute to those soil nitrous oxide emissions. People are interested in soil nitrous oxide emissions because it's a potent greenhouse gas. It's 265 times more potent than CO2. And it also tends to hang around in the atmosphere for an extensive period once it's up there. And the only way that you can remove it from the atmosphere is if it interacts with ozone. So it's not just a greenhouse gas, it's also an ozone depleter. So when we apply nitrogen fertiliser to the soil, what proportion of that might be emitted from the soil? Well, the international default factor that is used by the IPC is 1%. So for every 100 kilograms of nitrogen that we apply to the soil, it's assumed that 1% of that will be emitted as nitrous oxide. That doesn't sound like much, but when you start incorporating that value into life cycle analyses that calculate the carbon footprint of our agricultural products, it can have quite an impact. Here's a case study from Cunderdin, and the life cycle analysis was done by a colleague of mine at Curtin University, Waddell Biswas. And in this instance, when we used the international default factor, you can see that we calculated that 487 kilograms of CO2 equivalents would be emitted per tonne of wheat. And 36% of that was due to soil nitrous oxide emissions that were directly emitted as a result of applying urea to the crop. So we were curious, is this 1% realistic? This 1% value, which had really been derived from studies that have been conducted in the Northern Hemisphere on different soils to ours, utilising different fertiliser technologies and rates to what we use here in Western Australia. And thankfully, thanks to funding from the former Australian Greenhouse Office and DPIRD, under the leadership of Dr. Bill Porter, who I'm very happy to see here today, we were able to acquire some automated chambers to enable us to get out into the wheat belt and measure those soil nitrous oxide emissions and test if that 1% value was realistic here for WA cropping soils. And we embraced this new equipment. We started off out at Cunderdin. We then moved to Wongan Hills, and later further north to Barton, where we worked with the Levy group, and you'll be hearing from Stuart later this afternoon, who was instrumental in the site that we worked at Levy, and also Fran Hoyle. So, what do we find? Now I'm going to walk you through this graph. Don't panic. So here, what we've got here is a graph from the Cunderton research site where we measured nitrous oxide emissions, in this instance for 3 years, but it was actually 4. And what I've done is plotted grams of nitrogen per hectare per day lost over time. And over the 3 years at that site, we grew wheat for the first 2 years, followed by canola. And you can also see that we had 2 treatments, no nitrogen in the hollow circles, and plus nitrogen in the solid circles. And we'd applied either between 75 and 100 kilograms of nitrogen per hectare each growing season, and the triangles show you when, at what time of the year that we applied those. Okay, so it's a couple of things to point out here. You can see first of all that the daily emissions, they're not static, they're not the same day in and day out. So thankfully we had those automatic chambers, otherwise we could have missed that. The other thing you might notice is that the losses don't seem that large. They range from less than 0 through to 7 grams of N per hectare, which is small when you think about the fact that we were applying up to 100 kilos of N per hectare over our growing season. And you might have also spotted that there doesn't seem to be much difference between the treatments with and without the nitrogen fertiliser. And the other standout thing you might notice is that we got our highest emissions in response to summer rainfall events, rather than in direct response to nitrogen fertiliser applications. Now, when we added up all of those daily emissions, we could come up with an annual loss, and where we had both a 0 nitrogen fertiliser treatment and a plus nitrogen treatment, we could calculate an emission factor. And you can see that the emission factor that we calculated out at Cunderton was definitely much less than that 1% value that is used internationally. Was that a fluke? Was that just Cunderton? No. When we went to Wongan Hills and again to Barton, we again reported very low annual rates of nitrous oxide emissions, and again when we could calculate emission factors, they were still consistently low. So what? Well, first of all, thanks to the work that thanks to 3 continuous national research campaigns, of which Western Australia was part of each and every time, Australia no longer uses that 1%. The latest value that is now used for estimating direct nitrous oxide emissions from the application of nitrogen fertiliser to land is now country specific and varies depending on the production system. So for dryland agriculture here in Western Australia, we use 0.41. The other so what is, it means that we can more accurately calculate the carbon footprint of our agricultural products, which has huge implications when we come to export our grains. So going back to this slide that you saw earlier, this was what we calculated when we used the international default factor. When we use our site-specific data, you can see now that rather than 487 kilograms of CO2 equivalents per tonne of wheat, we now have 304, and the direct soil nitrous oxide emissions now only contribute 9%. What if you still want to mitigate, the emissions are low, but what if you wanted to still lower them even further? It's tricky. Internationally, the approach that people take is to lower their nitrogen fertiliser imports, or they strategically time their nitrogen fertiliser applications to better match crop demand. But what if your emissions are largely in response to these summer rainfall events? We hypothesised that those nitrous oxide emissions that we were seeing in response to the summer rainfall events was largely as a result of the nitrification process. The soil was dry, it rained, it increased mineralisation of the soil organic matter, and then that nitrogen, mineralised nitrogen was then subject to nitrification. And in the absence of crop growth, because this was occurring over the fallow, that nitrogen was then available for soil microorganisms such as the nitrifiers to emit nitrous oxide. But we also knew from the scientific literature that if we were to increase the soil pH by liming, especially on our acidic soils, then there might be a chance that we could also decrease soil nitrous oxide emissions from nitrification. So we thought we'd test that hypothesis. Off we went to Wongan Hills. And in this study, what we looked at was the impact of liming the soil from liming a wheat rotation on nitrous oxide emissions. So here we have the daily nitrous oxide emissions with time from over a 2-year period where we had wheat followed by wheat. And then you can see, Chris, you'll be happy that when we applied our 3.5 tonnes of lime, which we calculated thanks with the assistance of Steve Carr up the back there somewhere, we did increase our soil pH, and to a level that we hoped would mitigate the nitrous oxide emissions from nitrification. Like Cunderdin, again we observed these elevated nitrous oxide emissions over the summer rainfall, over the summer-autumn period. But did liming actually decrease the nitrous oxide emissions during those events? You can't tell. This is a terrible scale. I know this. So let's blow it up. All right, so here now we've got micrograms of nitrogen being emitted per metre squared per hour. This is the beauty of those automated chambers. We could now look at hourly fluxes. So these are the emissions we saw following a 45 millimetre rainfall event one summer. You can see as soon as it rained and the chambers closed, we saw a spike in the emissions from both the limed and the non-limed treatment. But what also occurred is from the lime treatment, the emissions started to decline much more quickly than the no-lime treatment, so that when we added up the cumulative losses from that particular event, they were significantly less from the no-limed than the limed. And so over the period of that study, we had 5 summer rainfall events, and 79% of the total emissions occurred as a result of those 5 summer-autumn rainfall events. From the wheat rotation, liming definitely decreased the extent of those losses. But what I didn't tell you was we also had a lupin wheat rotation, and in that case, liming had no effect on the emissions. We went back into the lab at the same time. We took soils from that site, and that lab-based study also informed us that when we increased the soil pH by liming from those particular soils, then we only saw a decrease in soil nitrous oxide emissions when those emissions were associated with nitrification, which is what we expected. So to wrap up, some concluding statements. I do have my timer here. Nitrous oxide emissions are relatively low from our semi-arid cropping soils in Western Australia. We've been really fortunate to have this investment here in the West, but when it comes to other semi-arid regions in other parts of the world, I don't believe they've been so fortunate. And as I hope I've demonstrated today, having accurate data is important for national greenhouse gas inventories, but also when we come to calculate the carbon footprint of our agricultural products. As much as I like getting out in the field, and my team does, we can't be everywhere all the time. It's just too expensive. Plus, one day I would like to retire, Lynn. So we're going to have to get better at modelling. And at the moment, my experience is simulation models just aren't doing very well with our flux data, and that's largely because they can't seem to be able to model these highly episodic events. Which leads me to this final point, that the regulation of soil nitrous oxide emissions following summer and autumn rainfall events is really not fully understood in our region and definitely still warrants further attention. So is it time to return to the lab? Okay, thank you.
Thank you, Louise. Our next speaker is Director, Institute of Agriculture, University of Western Australia, Hackett Professor Kadambot Siddique. Professor Siddique has more than 35 years of experience in agriculture research, teaching, and management with an extensive network of scientists within Australia and overseas. Siddique has a national and international reputation in agriculture science, especially in the fields of crop physiology, production agronomy, genetic and breeding in cereal grains, pasture legumes, and oilseed crops. Professor Siddique has trained numerous master's and PhD students and was awarded with the prestigious Western Australian Scientist of the Year in 2023. Ladies and gentlemen, please welcome Hackett Professor Kadambot Siddique.
Thank you, Hira, for that kind introduction. Good afternoon. And it is my great pleasure and privilege to be here today to help celebrate Professor Lynette Abbott's incredible 50 years milestone at the University of Western Australia, but also you heard from others in Western Australia, Australia, and internationally. So when did I meet Lynn first? Precisely 43 years ago, August 1981, when I came for my PhD. Lynn was very quiet, shy, and just moves to the university faculty tea room. That's where all the heated debates happens. So you see the debate between the late Professor Alan Robson, Bob Gilks, and about the phosphorus. And so we, as a postgraduate student, sits there. Lynn doesn't lean any side. She just smiles, and a big coffee mug. She walks from the club to the soil science building. I had not much to do with Lynn, but I just look her and admire. She talks about the publications in New Phytology. I don't know what is New Phytology at that time. Then I finished my PhD and, in 1984 and went to the then Department of Agriculture, where I started working on wheat and later on legumes. Occasionally I see Lynn, particularly in various meetings at the DPIRD or the Department of Ag, then at the Ag Institute. That's where I met Don, Lynn. When I came back to the university in 2000 to lead the Center for Legumes in Mediterranean Agriculture, I haven't had a lot of friends in the university at that time. all these academics always look, who is this guy coming and rule us? So Lynn was one of the first persons to come very quietly. Probably normally she comes around 5-ish and said, how are you settling in? If you need any help, you can count on me. So our friendship continued. We have done quite a lot of, particularly looking after international students. So when I had some difficult issues, not necessarily coming from me, coming from some other academics, for example, without telling the name, a couple of students, after 1 year, they sort of realized that's the wrong supervisor way to go. So I used to go to Lynn and say, look, can we just help these students? So Lynn said, can you be a co-supervisor? I said, that's not the main issue. The issue is to help these students. With that, Lynn has helped a number of students from overseas. They successfully completed the PhD and gone back to their home countries, including Malaysia and so on. Then I met Lynn in various capacities at the heads of the school. So for the CLIMA, I has to interact with the heads of the school. And then Vice Dean, we had quite a lot of interaction. And Lynn had made some important decisions to help the center to survive for such a long period of time. So that's the background information. My role today is to talk about Lynn's contribution for the past 18 years to the UWA farm. So I will stick with that one. Can you all see that? Okay. So the UWA farm, those who doesn't know, it is a 1,600-hectare mixed enterprise in Pingelly. And we have to operate commercially. What it means is that the university is not going to give money. If we make loss, it will be in the book. If we make any profit, so-called profit, it will be in the book. We can spend it on farm activities. There are about 600 to 700 hectares of cropping, 7,000 sheep, that varies depending upon. It's managed by a very able person, Tim Watts, and 3 casual hands. And we have got 2 committees. One is a commercial committee. Both committees I chair currently is largely led by some farm advisors, farmers, and the farm manager. And that's where most of the decision regarding the operation is made. Ashley Herbert is one of our consultants. The research committee is where I chair. We have got all the researchers from various parts of the university, and Lynn has been an active member of that committee for a long period of time. So the primary aim is research projects, field trips, teaching purpose, and interacting with the wider community. Not only UWA, but also we have DPIRD and CSIRO and Murdoch in that. And we have some accommodation available. Currently, we have about 15 to 20 active projects. So it's a multidisciplinary research and innovation activity we call Best Practice Farming System Project. And Graham Martin, you're here somewhere? Not today, so I'll talk about that later. So the main activities are mitigation on farm greenhouse gas emission. I'm glad Louise Barton just mentioned. She has a project actively there now, both from cropping and also from the animals, mainly sheep. Adaptation for the changing climate. I think you all know that. Australia is the driest human-habited continent in the world, and it will continue to so. So that's a major activity we are working on. And profitable and ethical animal production system, particularly animal production system, so that's going on. Restoration of the ecosystem and biodiversity, that's where Lynn comes. We have about 30% of the farm, of the 1,600 hectares, we are not going to crop because it's not going to be profitable in and out, so we're leaving for the restoration part. Of course, being a university, education, the students, the community engagement, the 3 communities we work are Pingelly, Shire of Pingelly, Brookton, and Narrogin. So what we like to see is that if you like the Californian model, Silicon Valley, etc., we want to have an impact of the farm, at least the 3 neighboring shires and beyond. And that's our aim, and we hope we'll achieve that. So Lynn is a very, very strong supporter of the Institute of Agriculture family. I think that's because of her long-standing position here over the 50 years with a lot of knowledge around the place, what to do and what not to do. And Lynn never upset anyone, so I have learned a lot from her as well. And she participates in the numerous public events. What I see in the transformation in Lynn since I left to the Department of Ag, most of her work was largely confined to the glasshouse, Kevin Snowball, and Alan Robson and all that. And when I came back, already you can see the transformation where Lynn moving into agriculture area and going and talking to the farmers. And when I was leading the CLIMA, we shared quite a lot of exhibitions and things where she brings the soil biology, microscope, earthworms, and attract all the kids as well as the farmers in that area. So that's the transformation I saw within a period of 10, 15 years. Darren Field Days and also the 2050 project, which I will mention a few more. And she also served as the deputy director of IOA when Graham Martin went to Oxford for a one-year study leave. And again, it has been very good. We catch up every week, talk about ideas and what to do, etc. Again, that has been very valuable for me, Lynn. So building ecosystem and biodiversity, and this is where the mixed tree plantation at the farm Ridgefield, this was actually initiated by Graham Martin and support from Bruce Ivers, a farmer who has been an incredible champion. Is Bruce around today? No. And then Lynn taken the oversight on that. This is again, she's going into different territories. She was not scared of going and working on some incredibly difficult projects. Then the other one is Kids to the Country program. And this is where strategic tree planting by school students and student volunteers since 2012 continued for probably 10 years. And if I can show footage, is it work? It's working. So here you can see that we chose a very difficult part of the landscape. There's a natural creek in the property, and these are all planted by school kids under the guidance of Lynn and of course Bruce Ivers and others. And this is really an incredible, and when you go there, you will see how much that has been achieved. The next one is pasture restoration funded by Wheat Belt and Aram. And then Lynn got the ability to go and talk to various people who are, sometimes you may not get a lot of money, but Lynn doesn't worry. Even if it is a small money, she's happy to work on that. And that's in collaboration with Philip, Barton, Lynette. Phil, here? Not here. All your friends are not coming, Lynn. Now look at this here, 2015 on my left-hand side or your right-hand side, and 2020. That's the restoration of the salt-affected land by introducing mixed perennial and annual pastures. So normally, we as good researchers, we go to the best land. And this is the most difficult land, and you're doomed to fail. But Lynn was not worried about it because she doesn't care about the publication, because if you fail, then you won't have a publication. The funding is not from some of the funding bodies who really insist it has to be successful. Another one, Smart Farms Demonstration Site. So here, this is again funded by National Land Care Program, an Australian government initiative, intensive regenerative practices establishing permeable biomass walls and trenches. Trees were previously planted by Greening Australia, and by the Greening Australia as part of the Australian government 20 million tree program. So again, you can see how things has been restored on those part of the landscape. Soil restoration, organic amendments, now comes to her own baby. That is restoration of soil conditions using permeable biomass barriers. I'll show some photos on that. So you can see here, fast-track restoration of the soil conditioning using barriers, putting some trenches and burying them. And then on the other side, you can see some of the trees now going reasonably well there. Examples of the biological amendments include straw, that is the straw returning, compost, biochar, worm juice, and wood vinegar. So Lynn is very active there. She finds a great passion in driving all the way with volunteers and students in restoring that land. The biological community established, basically roots proliferate into the cavity. That's what her hypothesis was, and that's what she's testing in this difficult part of the landscape. So here is the drone footage of the UWA farm Ridgefield, where that particular work has been going on for the last 10 years. And I'll just see the footage if it works. And then, of course, you heard about the soil app. This is, again, another area which she hasn't got a lot of expertise, but then she went and got some expertise to put into app, includes ebook, podcast, and 7 soil health animations. Again, very popular internationally. A lot of people come and ask to have a go at it. Come some serious projects, soil carbon, future carbon project. This is the Department of Primary Industries initiative. In the first round itself, Lynn, you were able to get, although small funding, but it is a 6-7 year project. Grazing into the future for soil carbon sequestration and building soil health. That is what the title of the project. Here, she's tying the potential for combining perennial and annual pasture species in medium to low rainfall zone to increase soil carbon sequestration. Again, not a new idea, but can we test that in some of the difficult part of the farm? So the annual pasture species, subclover, balancer clover, and annual ryegrass, and the perennials were tall fescue, phalaris, and rhodus grass. So Lynn went and started planting some of them in mid-winter. I said, just you have to wait till some more time, till the soil warms up, but they went and did that. Huge, huge impact. So basically the last 50 years has been incredible, trained numerous students, learned a lot of basic things from the laboratory to the glasshouse, into the field, and still continuing vigorously. And sometimes I ask Lynn, you need to take some break, a rest. Then she says, you are not taking any break, so how can you advise me on that? But it has been an incredible experience working with Lynn as a mentor, as a friend, and whenever there is an issue, it's worth going and talking. The other thing I notice is that I'm very organized, whereas Lynn can do things at last minute. So you have got a project, $5 million project. We all sit and work together, and what's happening? And very silence, but midnight, 2 a.m., 2 p.m., just works, and then I have submitted the proposal. So I don't think I can do that, but I really take salute to you for some of the work. Thank you very much, and I think I served some time.
Now we will go ahead with our symposium with our next speaker, Stuart McAlpine. Stuart McAlpine is a 4th-generation farmer in Western Australia. He is a co-founder of Levy Group and has served as its inaugural president in 2007. He is a non-executive director of Perth NRM and chairs Region WA. A practitioner in soil and regenerative agriculture, Stuart was named a soil champion during the international years of 2015 and became part of the Northern Agriculture Catchment Council's honor roll in 2016. He is also a regular speaker at the Western Sydney University Soil Biology Masterclass. Please join me in welcoming Stuart McAlpine.
Thanks very much, Hera, and what a great honour to be here today for Lynn's celebration, but to be with so many other colleagues and that I've worked with over the years. And I know Lynn said she didn't want to make it all about her, but it is really important that we get out and work with our farmers, and we've certainly been blessed with the Levy Group having so many of you wonderful people that are out there today working out on the coalface with us as farmers, trying to understand our soils and how they function. So it's important to inspire curious people and crazy people like me to take some of that research and perhaps even send some research back to you guys. So for me, Lynn's foundational work on soil biology has been particularly inspiring. Lynn has always encouraged me to embrace soil biology, which has been overlooked for far too long as a critical element of soil health. And I know for me, for a while, it was quite a lonely space when I really went hard into soil biology, and Lynn was always a great person to talk to and be passionate and keep my head up, so it was great, and thanks for that, Lynn. We've had the... Ooh, why isn't it showing up? Oh, here we go. There it is there. That was... we're missing a slide, are we? No, here we go. So this was taken, I think, back in 2013 up at Leby, I think, Lynn, one of your many visits up into the area. Many times we've spoken together. But it's just a great example of how Lynn's work has inspired people to be interested in soil biology, and so much so that we did have a soil biology site at the Leby group. So I'm going to talk a bit about my journey, which is going to be heavily about soil biology. My journey into farming began when my father was diagnosed with terminal illness. So at the age of 14, I then decided I would return to the family farm and forgo any formal academic training after completing Year 12 at Guildford Grammar School. I relied on curiosity and innovation, becoming an early adopter of precision farming, high fertiliser rates, no-till practices. We were very successful high-input farmers, and a lot of that goes down to the innovation of my father as well. After my father passed away, I co-founded the Leby Group in 1997 to foster research collaboration in our very isolated region. This was a game changer, connecting us with leading researchers and institutions from the State Agriculture Department and its various titles over the years. CSIRO, agribusiness companies, and with assistance from the GRDC and other institutions, we have collaborated on research that values farmers as key partners. One of our earliest projects was a long-term trial on my farm with the then UWA professors Dan Murphy and Fran Hoyle. I see Fran's in the crowd, focusing on soil health and soil biology in particular. Some of the great work that Dr. Louise Barton has just talked about, which was actually on that site, was also really important, and there's so many of you in the room, I can't mention all of you. So I farm at Bunteen, which is pretty isolated, 5,000 hectares, but I just wanted to touch on this because this was sort of where I got to with the Leby group. Was we were doing all this fantastic research, and productivity gains, I think, through the 90s were sitting at about 3.5 in Western Australia in our broadacre farms. They've dropped to about 1.5 now. But we were getting better, but they didn't seem to be compounding out, and we were just spending more money, taking more risk. And in the 2000s, we experienced 3 severe droughts, which led me to question our high-input system. Despite increased productivity, profitability wasn't improving, the risks were growing, and I realised that soil health was probably declining at a similar rate to our productivity gains. So that's in Canada, and it's quite a good one, but it just shows you the green bit is the farmer share. So you can see we're spending so much money to make similar sort of returns. I sat down because I remember in 1975 we averaged 2.6 tonne to the hectare, 13 bags for those that remember that, in that time, and I thought, you know, we should be able to apportion those productivity gains that we're seeing on that yield in a good season, and on a good season we should be able to see that compounding productivity over time. So I've always kept this running over time, but I think you can see that you know, if we were to put together all the variety increases and all the placement and all the things we do, you know, these are the sort of things that should be achievable. And I sort of theorised that at the same time, there was one component we were spending a lot of money on the physical and chemical side of our soil, but really the too hard basket was definitely the soil biology. So I started getting very interested in that and trying to find that sweet spot in the middle there where everything starts coming together. So this insight prompted me to explore new approaches. Around this time I was introduced to a biostimulant called Best TM Agriculture. Although sceptical at first, initial trials showed promise. I began observing remarkable changes in my soils and others, as well as in pastures and plant health. Rather than lay off workers during the droughts, I decided to undertake broader evaluations of the product across Australia. In 2009, I attended a lecture here at the UWA University by Professor John Crawford, who's now Emeritus Professor of Sustainability and Complex Systems at the University of Glasgow. His seminal work, Interactions and Self-Organisation in the Soil Microbe Complex, profoundly impacted my understanding of soil biology. His research illuminated how microbial interactions drive self-organisation in soil, enhancing its structure and function. This lecture was pivotal in inspiring me to trust and harness soil biology, reinforcing the observations I was making on my farm and shaping my journey towards biologically driven farming systems. This led me to delve deeper into soil biology, uncovering its potential to enhance soil health and system resilience. And I think it's quite timely that the Haggertys have just been put forward as Australians of the Year, and they call their system natural intelligence farming, and I think it really works in with some of that stuff that Professor Crawford told me about all those years ago. So this is what really gets me excited. I mean, this little wheat plant was planted 5 days, hadn't even emerged or started photosynthesising, and yet you can see the colonisation of the microbiome and the strong rhizosphere already. It's sticky and there's just amazing stuff going on. So the challenge for me was always to understand what that actually meant once I activated that, and it's management that makes that work. It just doesn't happen by itself. So one of the first indicators of change from observing was improved control of rhizoctonia and improved herbicide efficacy during the drought. So I started in 2006, which was a pretty bad year. I was able to measure yield improvements of around 30%. Roots were developing in a strong rhizosphere, and the A horizon was moving deeper at about 25 millimetres per year of application. Oops, go back. There you go again. There's another little nice Medusa type roots on a cereal plant that's probably about 4 or 5 leaf stage. Then a soil testing contractor remarked on how much softer my soils were compared to others after 3 years in the program, and I hadn't done any deep ripping for about 5 years. In one instance, I had a farmer that had applied product and observed improved weed control side by side, but at the other end of the paddock there were no weeds to be seen, but we could feel that the soil was much, much softer underfoot where the product had gone out and the roots were going down deeper. We had tissue tests consistently showing higher nutrient levels, even with reduced fertiliser applications. I noticed that while my crops might have started a bit slower, as I pushed the system and the biological system harder, they always finished strong. This was due to plant exudates feeding the soil, feeding the microbiome and developing healthy roots within a robust rhizosphere, which in turn supported the crop through the growing season. The result was healthier, more resilient crops with superior grain quality, particularly in challenging seasons. And you can see, the dark exudates in the soil in the lupin crop and the old marker there and how thick those lupin roots are as well. This one here is I observed in 2060, in a particularly frosty year, that I wasn't getting all the frost that my neighbours were getting, and I was measuring up to 5 degrees difference in soil temperature during those frosty conditions. This isn't from my farm, but you can see there's a green paddock there. That was a treated paddock with active biology compared to the rest, so there was just a lot more respiration and activity going underneath the ground. Consumer feedback reinforced the benefits. A dairy farmer using my lupins reduced his feed rations by 30% after switching to my lupins. Similarly, feedback from Dirty Clean Food on my cattle. I only supply once or twice a year, and my cattle were consistently best in class for carcass quality, despite coming off my paddocks end of April, end of May after hot, dry summers. The marbling scores were unbelievable. Financially, my consultant noted that while my yields might not always top the charts, my risk was significantly lower, translating into consistent profitability over different seasons, and we'll see some more data on that soon. So I've had to do a lot of my own trial over the years because I had to understand what activating this soil biology meant. So I've done lots of trials with fertilisers, rates, different biostimulants, the list goes on. Pesticides is an interesting one, but I don't have enough time to talk about that today, but love to talk to anyone about that at some stage. Initially I was cautious about reducing inputs. To mitigate the fear of missing out, who wants to miss out when you go to the pub and talk about your yields, I implemented zero input trials alongside biological stimulation to quantify the maximum I would miss out on, or so I thought. Over time, these trials showed that even with no inputs, yields were often close to those achieved with traditional inputs, thanks to efficient nutrient cycling, Research is lacking to understand where an oversupply of nutrition reduces the biology to activate as well. So, research question for you people out in the room. I did put in a trial about 15 years ago with GRDC on that one, but I haven't seen it yet. So here's just an example of some of the extensive trial work I've done on farm. Lots of different rates of traditional chemical-based fertilisers, more biological ones, and some liquid treatments. But I just want to focus on a couple. The highest one that delivered the most profit had no fertiliser but a calcium-based product, and the highest yielding one also had a calcium-based product, the AgFlow yield, at 2.02. So there was some good data in that trial. The control, which actually had nothing, was also made more money than the other one. But if we go on to what that actually means at a farm scale when you're looking at economics, you know, there was an extra $16 a hectare return over the 3,300 hectares of wheat. So that made an extra $54,000, and this was done a few years ago now. But, you know, more importantly, the high rate of fertiliser was, you know, $60 a hectare more invested. So if you put that into perspective, that's an extra $150,000 capital at risk. And in an environment where we're seeing such variation, we need to really manage, you know, how we spend that money and where we need to spend it. So, and then this year, I was able to, following sort of a really bad drought, we had our worst year ever in 23, and similarly challenging start in 24, I planted my entire program without any starter fertiliser this year. This decision was backed nearly on 20 years of on-farm research. Even with reduced early rainfall to activate the biology, the results were impressive, demonstrating the system's resilience. And thanks, Nick Ayres. If any of you want to work with a great young agronomist, Nick Ayres I can recommend. But this is a bit noisy, so it wasn't a great trial, but you can see the nil control still delivered some pretty healthy yields of sort of averaging around 4.3 tonne to the hectare with no fertiliser at all. So that's not what I advocate, but I know that I can do it when I need to, and I still putting back on the nutrients that I export in my grain. Pretty happy there. In my journey towards more sustainable farming, reducing reliance on high rates of fertiliser has proven transformative. By allowing plants to regain control over their nutrient intake, I've observed a significant shift in how biomass is managed. This change has improved our ability to navigate dry periods as plants naturally adjust their growth according to available resources. During hot, dry conditions, excessive nutrients that would typically fuel undesirable growth are no longer a burden, allowing water to be used more efficiently. So this crop here, this is 22, which was our most profitable and highest yielding year. I had numerous paddocks yield 3 tonne of canola with only 17 units of applied nitrogen. And I think, you know, you need about 80 units of nitrogen per tonne of grain to produce a tonne of grain, so that's 200. So that nitrogen's still got to be there to grow that crop. But it's a testament of getting that biological system going. Healthy soils have also brought about a remarkable reduction in the need for chemical inputs such as insecticides and fungicides. The improvement in soil biologies have enabled me to eliminate many of these chemicals from our system almost immediately. While I remain open to using them when absolutely necessary, such instances are rare. A thriving, balanced soil ecosystem provides a natural resilience that helps protect crops from pests and diseases, reducing dependency on external inputs and fostering a more self-sustaining system. This shift not only enhances the farm resilience but also aligns with a broader vision of farming that prioritises long-term soil health and ecological balance. So I want to talk about plant exudates and feeding the microbiome, because I think this is a really important area for research and getting to understand more. So this is a treated and untreated, and you can see the darker soil from the exudates being pumped in there, and a typical example of what we see with the roots pushing deeper into the profile with an active microbiome. Understanding the dynamic interaction between plants and the microbiome marks a transformative step in managing soil health, especially in the dry West Australian climate. At the core of this relationship lies photosynthesis, the process by which plants convert sunlight into energy. A significant proportion of the carbon fixed during photosynthesis, often 20 to 40%, can be allocated below ground as root exudates. These exudates, comprising of sugars, amino acids and organic acids, serve as a primary energy source for soil microbes during a range of soil building processes. Soil biology fueled by these exudates play a crucial role in nutrient cycling, converting organic and mineral forms of nutrients into plant-available forms. Unlike conventional systems reliant on water-soluble fertilisers, biologically active soils allow plants to access nutrients on demand, enhancing their ability to adapt to variable climatic conditions, including extended dry periods. Beyond nutrient cycling, soil microbes contribute to soil structure. Fungi and bacteria produce substances that bind soil particles into aggregates, improving water infiltration and retention, particularly in our soil that are very sandy here and prone to rapid drying. The interplay between soil biology and carbon sequestration is another key function that needs a lot more research. As plants fix carbon, a portion is stored into the soil through microbial processes, enhancing soil organic matter. This further improves soil resilience, enabling it to better withstand climatic extremes such as drought and heat waves. This is a very old slide of Lynn's, and she may well have updated it, but this was really useful for me because I understood that some of these changes were going to take time in my soil, and it was work like this that allowed me to be patient, particularly when the economics weren't affecting me. It's really important to, transitioning to a biological active system does take time and requires consistent management. Practices such as reduced tillage, diverse crop rotations and the use of biostimulants enhance microbial diversity and activity, fostering a self-sustaining system. Now, I sent some DNA samples away to the US a few months ago, and it's come back, and it's quite interesting. And I'm not going to focus on the results in particular, but I think some of these emerging tools that we have, and as we start to understand the function of different parts of the microbiome, it's really quite exciting to take our soils to a new level. A lot of these results align with my testing that I've done, in fertiliser efficiency and stuff like that. And for instance, they're picking up a lot of functionality around fungicide protection and insecticide, nutrient cycling, which are things that I've been seeing. So, I think we need to keep testing this, but I think it's quite exciting because, one, we can see what is missing and then maybe add it if our soils will take it, which is uncertain. But I think if your focus is on stimulating what biology you actually have in your environment first, then you've got more chance of introduced beneficial biology of surviving. So, and another great thing for the test, it does actually measure a lot of the pathogens as well. So I think this test is about $600 Australian. So really interesting stuff. I think, once we start coupling this data with, what are the total nutrients in our soil, we start getting to a system where we can be a lot more efficient. I've been working, so this is an early test I did on biostimulants, and as we continue to refine our understanding of soil biology, I'm confident that we can push these systems to new heights through ongoing trials and collaboration. We're unlocking the full potential of the principles of regenerative farming. And this is a really easy way to ascertain whether a biostimulant is actually working in your soil, because there's a lot of them out there. DNA testing might be another way to do it, because once you activate the soil biology, I believe a lot of the rest of the benefits come from your management. So, you can test products pretty easily, I reckon, in your soil, in your environment. They may vary from season to season, so you need to retest every now and then. Again, there's a lot of detail in here that I cannot explain in this period of time, but this is some stuff I've been doing with Downforce Technologies to enhance landscape management by integrating satellite imagery with ground-based data to create higher resolution digital models of land areas. This data fusion approach enables precision monitoring of soil organic carbon and other natural capital assets at a 10 square metre resolution, providing actionable insights for a sustainable land use and carbon sequestration strategies. So if you think about that, 10 square metres, that's the equivalent of 1,000 data points or 1,000 tests in a hectare. So you can start to see how some of this stuff can really, as we get better and it's better and more useful, we can really start analysing our system and much, much better. This is one of moisture. Interesting enough, in this, the big improvements have come from lupin and canola crops in carbon and moisture retention. Happy to talk about some of the reasons why for that later, but I don't want to, I've got to keep moving. So I think satellite technology plays a crucial role in modern agriculture and monitoring practice change at the landscape level. It provides high resolution data scale that is both objective and consistent. In addition, satellite monitoring supports climate resilience by analysing the effects of extreme weather events and guiding adaptive measures. It is particularly valuable in tracking carbon sequestration and other ecosystem services, aiding in sustainability reporting and at much more affordable cost and compliance with environmental standards. By integrating satellite data with ground measurements, this technology serves as a powerful scientific tool, helping farmers, researchers and policymakers make informed decisions to enhance productivity and environmental stewardship. So I think there's some tremendous opportunities for research in this area. So look, soil biology has been transformative on my farm, improving soil health, crop resilience and profitability. By embracing a more holistic approach, we're better equipped to manage risk and adapt to changing climate. Since 2011, as mentioned, I've been presenting over at the Western Sydney Soil Biology Masterclass, and Professor Raj Singh is another great mentor of mine. But I love going over there each year and seeing the new science. I go, wow, that's what I've been observing on my farm, you know, that explains what I've been seeing. So it's an exciting time for soil science, and I'm grateful to you, Lynn, and to all you researchers in the room that have inspired me and guided many others on this journey, or on all of our journeys. So together we're building the foundation for a more sustainable and resilient future in agriculture. I encourage more collaborative work to understand soil health principles in regional farming systems, as these farmers ultimately provide a food that sustains the soil. Thank you. And this is just a quick, this is some of the stuff that Lynn's got up there, free access on soil health. She's got a great stuff out there that really, if you want to understand it a bit more, great stuff, Lynn. Thank you.
Thank you, Stuart, for bringing us experience from the farm. Now I'm very delighted to invite Principal Research Scientist, CSIRO, all the way from Adelaide, Dr. Mark Farrell, who has joined us today to celebrate Lynn's 50 years of research and soil science, along with the development in this field. Based in Adelaide, Dr. Mark leads the biogeochemistry team. He also holds a position of Adjunct Associate Professor at UWA. The focus of Mark's work is the interplay between carbon and nitrogen in soils, particularly in Australian agriculture system. He works from the mechanistic scale of carbon and nitrogen cycling using many advanced technologies to broadacre agriculture and environmental system scale. His work is funded by many national stakeholders, including Grains Research and Development Corporation, South Australian Grain Industry Trust, Cotton Research and Development Corporation, Department of Climate Change, Energy, Environment and Water, and Forestry Corporation, New South Wales. He collaborates widely with agronomists, ecologists, and modelers to achieve impact beyond discipline-specific advances. Dr. Mark Farrell.
Thank you, everyone. I recognise I'm the last speaker before the person you all would actually like to hear speaking, so I shall try and keep this brief. I also will give a bit of an apology ahead of time. I was given a bit of a licence with that title to go super nerdy into soil carbon biogeochemistry, so it is probably the exact mirror picture to what Stuart was just wonderfully presented from on the ground. This is the other side of the story, and the great thing about events like this is that both sides come together and hopefully something is found in the middle. So, mechanisms of soil carbon storage. That's a changing topic in and of itself. I'm going to take a step back first. Slide that I forgot to rebrand from a GRDC talk. Apologies. I'm not going to talk about nitrogen much. I work on carbon and nitrogen almost all the time together. Very rarely do I separate them. I'm mainly talking about carbon on this occasion, but nitrogen's worthwhile keeping in there for the moment because it when it usually follows carbon and then it doesn't, and that's where the interesting stuff happens. I'm going to leave that as a side point, but it's just something to keep in mind. So in agricultural systems, carbon pretty much only comes from two places. You can either bring it in or you can grow it yourself. And given the amount of land that agriculture covers, bringing it in doesn't work everywhere. Indeed, it doesn't work many places in Australia. So if you want to build carbon, you need to grow more of it yourself. Also, from a climate change perspective, if you're bringing it from somewhere else, you've not actually solved the problem because it was already out of the atmosphere in the first place. That's something that often gets forgotten. Nitrogen, on the other hand, fertiliser, organic amendments, nitrogen fixation and end deposition. Not so much end deposition in Australia, but a little bit. Exports of them. Crop, you take fermented carbon away in crop. If you go around harvesting stubble, you certainly take a lot away, and that's an interesting topic in of itself as other aspects of sustainability drivers are looking for carbon sources. And stubble is a major source of carbon back to your soil. Don't rob Peter to pay Paul. Nitrogen, and it was great to hear Stuart talk in a measured way about biology, that you still need fertiliser, but you build the opportunity for when you've had a really bad year to be able to gain reasonable crop out of the ground with little risk because you had that buffer there. Increasing work being done, work on legumes has been done for decades, but there seem to be some real strides in legume agronomy that potentially move us further away from being completely tied to the bag. That's probably about all I'll talk about with nitrogen. I'm going to take a step back. So, organic matter. Back in the 1800s, a fellow called Justus Liebig, we talked about humus. Humus being the source of life, the source of nutrients in soils. Around about the turn of the century, people wanted to find a little bit more about organic matter than it just being one thing. They recognised there were different fractions of it, and there, thanks to the wonders of ChatGPT, you have some pictures of what it thinks are humin, humic acid, and fulvic acid. It's probably about as accurate as any other description of them, to be perfectly honest. So for reference, I was doing my undergraduate in the early 2000s, and I was still taught about these things as this was how soil was. So organic carbon was then. So this should probably give an indication of how recent the complete shift in our understanding of what organic matter is has happened. So around about, well, building up to year 2000s, but probably acceptance only in the late 2000s, was an acceptance that a lot of organic matter in soils is stabilised rather than stable. That's the big difference. So the whole idea of those big macromolecular compounds up there is carbon is intrinsically stable because it's really hard to break down. That has more or less been moved away from entirely, and that turns the entire game on its head. Because rather than you having a view that it's relatively stable no matter what you do to it, all of a sudden if it's being stabilised, it becomes a bit more apparent that things that you do in your management might destabilise it. Or alternatively, there are things that you can do to stabilise it some more. As I say, this is quite an evolving feast because even now there are new things arising. One of the things that was pushed around the 2010s onwards, really great advances in how we're thinking about incorporating microbial processes into models was the idea that most carbon flows through microbial biomass and most soil carbon is actually effectively dead microbes. David Jenkinson, obviously, famously was pushing that a long time ago at Rothamsted, not long on this graph, but I think it was 1975, his paper about the eye of the needle, etc. But coming back to today, there's an increasing awareness that actually fermented carbon doesn't necessarily have to go through a microbe. It can just go straight out as a root exudate and bind by itself, which has implications when you think about how models work, if you've got two very different processes happening. So I would say it's still very much evolving. But to cover off on those three different types of stabilisation mechanisms, chemical recalcitrance is still important. I would personally say that we've moved too far in the 2010s towards it not mattering what the carbon was, it was what it stuck to. I don't think that's right either. Because physical protection, again, this is research that predates certainly my research career. being locked up in aggregates. So there is absolutely nothing stable about this carbon. It just happens to be in the microenvironment in the soil that makes it impossible to be broken down. And then lastly, which is where most of the attention had sat, certainly up until the last few years, really focused on mineral absorptions, the absorption of lipids, fatty acids, those sorts of things onto clay materials. Realistically, all those forms of carbon stabilisation are important. Soil type to soil type, environment to environment, management to management probably dictates which of those is the greatest factor in any one system, but it is safe to say that all those factors matter. And the trouble is, and why it is so damnably complex, even for somebody who has the privilege of playing with the toys that we have at CSIRO, to get your head around is no one instrument, no suite of instruments, measures all those things on the same scale in a useful manner. Visual observations don't cut it either. It's really quite difficult. And I think that's why there are some people who like to maintain the mystique of the brown fluff that is organic matter. I'd quite like to have a simple answer, as simple as it is for you've taken off this much N in your crop, therefore you need to apply that much N. I'd love it if we could do that with organic matter, but I think it is a fundamentally far more complex game to be playing with. So that's a brief overview of, I guess, the state of play in science in general. So where are we at in Australia? A few years ago, there was this thing called SCARP. A few people in this room will have connections at this point because it was a scary, horrible, big project that did amazing things. This is data from SCARP. It was a project that's led by Jeff Baldock. There were partners around the country, Fran Hoyle, Dan Murphy, when they were at UWA, were major partners, and I'll get to Sockham in a minute. There were 23,500 samples, I think, analysed in our labs in CSIRO in a period of 2.5 years from more than 4,000 sites, which, if you've seen the size of our lab, which probably about a third of this room, that's quite a frightening prospect in and of itself. Nonetheless, this served as effectively the first baselining of soil carbon across Australia. There was data from all over the place previously, plenty of it collected really rigorously in and of itself, but this was the first unified study. And as I walked into CSIRO in 2010, this was in full swing, and it scared the life out of me. But it's enabled us to say some things. There's distributions there of different amounts of carbon in the different soil types. Some soil types, as we know, are far more likely to hold on to more carbon than others. Perhaps unsurprisingly and almost disappointingly that it wasn't any different. Cropping and mixed systems typically have less carbon than pasture systems. 2 reasons for that. They return less carbon to begin with, and they typically tend to happen in places where there is less rainfall in the first place. Despite all that data that we got from SCARP, the first thing was climate, the second was soil, the third was management. So you're still very much driven by where you are and what you're dealing with. And a lot of talk these days about dealing with soil constraints. Sometimes you just have to accept that this isn't a soil constraint, this is your soil. That gets missed a fair bit too. Just one thing of note in here. So the sizes of the symbols on that map, they relate to carbon stocks, unsurprisingly very high in southern Victoria and Tasmania. But you'll spot a few in WA too. I think the winner, Fran, was one in WA. The highest carbon stock soil out of those 4,000 sites sampled was in Sunday, WA. So, yeah. The other big thing that SCARP did, it took us beyond carbon stocks, which, like I say, you could probably have drawn that more or less, except for the surprise of WA's winning sample. Trying to get a lot of that theory that I opened with on what's driving carbon stability and make it actionable and useful and applicable at scale. Most of those methods require 2 or 3 days per sample, maybe a week, multiple bits of $1 million equipment to do. They're just not applicable out there. Great to be nerdy in a ivory tower of absolutely no use whatsoever out in the paddock. So one of the things that really, a lasting legacy that emerged from Scott was the acceptance that despite its complexity, we can distill carbon down into 2 or 3 functionally different things, and there are methodologies that get at describing those different aspects of it. This is a slide most definitely borrowed from Jeff. One day I might update it, but he did such a good job with it. Fran, I think, is also a user of this one, Dan and others. But basically, there are several different ecologically relevant pools that we've separated carbon into. The top 2 aren't really in the soil at all, plant residues on the surface. And for somewhat historical reasons, I'm not entirely comfortable with. Anything greater than 2 millimetres doesn't count as soil, even if it's in soil. We'll leave that for another day. But once you get into soil, you have 2 useful size fractions that this is adopted internationally too. The MAOM that the Americans use a lot, again, is that it's actually 0.053 because of imperial measurements. But this cuts off at the sort of silt boundary between particulate matter that sits in the sand and organic matter that's associated with the silting clay at 50 microns or less. All that so far follows a quite predictable trend. Carbohydrates decrease, C2 nutrient ratios decrease, and resistance to decomposition increases. But one thing that has always been a problem in Australia, and frankly it's a problem in many other places too, they just don't recognise it, is this resistant organic carbon, this charcoal-like material. Bearing in mind that soot is charcoal-like material, we're quite happy to talk about this stuff being recalcitrant in terms of millennia. There doesn't need to have been a fire last week for there to be some charcoal in your soil. And yet most of the rest of the world, I've been to 2 international conferences this year, still don't seem to get this, which is astonishing. But those fractions are what we can separate, what we do at CSIRO, and it's been adopted fairly widely across Australia with variations on it, separate organic matter into. It's POC on the left. You can see on the microgram, on the electron micrograph there, it still looks a bit plant material. There's bits and pieces of stuff that are recognisable in there. The one in the middle, the HOC, which is roughly relatable to what is called overseas mineral-associated organic carbon, but again, there are complications with that direct comparison. Those are clay particles with lots of waxy stuff on them. That's the dead microbes I was talking about. And then lastly, you've got some charcoal pieces that, again, fundamentally look quite different. So these are pools that visually, using electron microscope, you can see look very different to each other. We don't do electron microscopy much. It's one of the things perhaps we probably should do some more of. What we do an awful lot of is NMR. I'll go into some of these spectra on the following slides, but ultimately the ones on the left have that large peak in the middle. That's the oil kill, the carbohydrate, sugars, cellulose. Those dominate that, and that's the highly decomposable material. The one in the middle, the humus-like organic carbon, or the mineral-associated organic carbon, if you want to be American about it, there's a much more even ratio between that and the alkyl, the peak on the far right. And then the ROC, the charcoal-like material. Now that peak looks like it's in the middle, but it's not. It's actually one of the smaller ones on the other 2. That's very different material indeed. That's the aromatic charcoal-like material. If you've been using NMR for as long as we have at CSIRO, you can build some wonderful pretty graphs. Those are the fine and the coarse fractions for, I think there's 1,200 different NMR spectra on each of those. That's several years worth of running the instrument non-stop. But there are fundamental differences between those 2 sizes. That's why the 2 sizes is actually quite important, and the clay aspect to this is important because it's the clay that is driving this. The one thing that you should know, however, is the variability that there is. Do I have a pointer on this? No. There. All right, I won't try that again. I will not try to use the pointer. The peak to the left on them both, the one with much more variation in, that's the charcoal-like peak. And you can see that that is highly variable both sizes. Charcoal's large, charcoal's small. There are other fractionation processes that use density. Charcoal can sink, charcoal can float. Fishing this stuff out physically is frankly impossible when you account for the small size of some of it. So we're always going to be stuck with trying to work out what it looks like down the end of an instrument of one sort or another. Those spectra are fairly incomprehensible, even somebody who stares at them fairly often, but thanks to stats, we can decompose them to something a bit more useful. And there you can see the fine material, the orange, and the blue material, and the coarse material, the blue, quite distinct in a Prince components analysis, driven by that alkyl and alkyl, and wide variation down that aryl, so that's the aromatic material. Make it simpler. Alkyl to alkyl ratio. Jeff Baldock, who used to run the group at Adelaide till his retirement, this was what his PhD was on, fundamental metric of decomposition in organic matter. Those two size fractions do a really good job of separating processable versus processed organic carbon. The trouble is, they do absolutely nothing for the recalcitrant, resistant organic carbon material. And that's why we end up being bound with difficulties in understanding how carbon is behaving in soils. It's the same, effectively, resistant organic carbon is behaving not too dissimilarly to how we treat inorganic carbon in soils. We accept inorganic carbon is fundamentally different from organic carbon, and then make it go away with acid and pretend it never existed. Probably a bit of a mistake, and there's plenty of people who are trying to square that circle, because inorganic carbon is probably more important in the function of our soils than we've recognised. But we can't make charcoal just disappear with a bit of acid. It's there. It's, in actual fact, in the Green Book, in the official method for measuring carbon, there's a section about how you're supposed to be picking pieces out before you make your carbon analysis, which again doesn't work because a lot of it is small. So I think this is just something that needs to be understood, that we have this carbon in there that doesn't play by the rule book. It's also not completely inert either. It is doing things. It's just really doesn't behave the same as the rest of it. Going back to data from SCARP alone, these, again, are a whole load more NMR spectra. On the left-hand side is coarse, on the right-hand side is fine. 0 to 10, 10 to 20, 20 to 30. And there's that alkyl to alkyl ratio that is fairly consistent in how it changes with depth and across those two different size fractions. Again, the sizes are quite helpful. But again, there's the charcoal peak. It's varied. It's, on average, about the same in every single one of those separations of samples. And that's what makes the trouble with the charcoal material. So I'll take a step sideways for a second. Some biochars are not too dissimilar to this, but they behave so differently to the rest of the carbon in your soil, and that's what this stuff is doing too. It's not contributing nutrients in the same way, it's not contributing water holding capacity in the same way. Has different interactions with the biota, but it doesn't behave the same way. Taking that data, and with the magic of mid-infrared, which I won't go into too much detail here, but basically a tool to try and turn those weeks and years of lab data into something that's a bit usable and quicker. I've taken the 20,000 or so data points that exist from SCARP from the spectra and basically made a mess of a good old-fashioned ternary plot because we've got the three things. There are distinct differences with depth in how the three pools separate out. So again, if I can have a simple message out of all of this, it's basically that the carbon isn't carbon, and this comes back to the problem of being able to say, well, you have 2% carbon, therefore this. Just is not. It will never be something you can say that to, unfortunately. I think it can be made much simpler than it has been. I'm probably making a dog breakfast of doing so right now. But at the same time, it is fundamentally far more complex than looking at things like mineralization rates. I've talked to that's mostly data from SCARP, which is the best data set we have in this country and arguably one of the best in the world, I would say. Particularly when you account for the fact that we have that information on fractions, which allows us to talk a little bit about vulnerability and at least infer some function to that. One of the projects that I'm involved with at the moment, I'm co-leading with my colleague Sinonica and Ratner, who's based in Canberra. Fran Hoyle leads the West Australian node of that project, is looking at carbon, is basically revisiting 300 of those 4,000 or so sites that we sampled in SCARP and trying to look at change over time. Before we get there, this was a best estimate from a postdoc who's now a research scientist at CSIRO, and he pulled together, this is quite an impressive bit of work, pulled together data from the soil databases across the country and basically synthesized a map and tried to get it at change over time. It's interesting that there is a very small increase on average yearly. I think that might well be one of those numbers that might be right as an average, but is wrong absolutely everywhere at any individual time. And that's why projects like SOCM, which is the project that we're working on right now, is so important. And we've basically looked at pulling together, trying to find the most representative 300 sites from those 4,000. Sadly, we had far fewer dollars to play with than we had with SCARP. The 300 sites in and of itself, that was quite difficult to work with because it's all very well choosing the 300 most different in a computer and going those are the most representative. Again, as Fran, Chris and others will attest to, that might be the perfect spot on the map, but you ain't having a sample from there. Can you find me another one? Or an email the next week. Yeah, we need to find these 10 new sites. It might well be around about now that we finally have the last sample out of the ground in New South Wales. And bearing in mind that we were only trampling 300 sites rather than the 4,000 in SCARP, you can perhaps understand probably one of the biggest issues with building a monitoring campaign is being able to go back to where you were last time. All this is on private property. Owners change, property names change, parcels of land get sold. There's real life gets in the way of doing any of this. And we could do it on research farms, but then you'd know an awful lot about a very small part of the country. So a real practical problem with soil monitoring is, and we fell headlong into this, even with some fairly well defined areas. So, having had my 1 minute warning, I'd better hurry up. Some very, very preliminary results. This is one of those horrible structural equation models that basically tries to say an awful lot of things in one place. Again, not surprisingly, the one at the top pointing in, the biggest driver of all, cumulative annual precipitation. The next task, and these are data, I should say, these are only data from South Australia and Tasmania because these were the only data that were processed when this one was done. But I don't think that answer is going to change too much. The challenge for us will be distilling what management information, basically normalising for climate, and then pulling effective management out there. Pretty hopeful we'll be able to do that. One of the big things that we've really focused on this time is trying to get much better management information than we had with SCARP, and we've had a lot more advances in remote sensing, etc., and much more chance at filling in the blanks as well. So to summarise, how carbon is stabilised and lost is has most definitely evolved. It's still very much evolving. Pinning real numbers to what that means for everything else is still interminably difficult. Even the suppositions of if you increase carbon by this much, you'll increase how much water holding capacity. Doesn't really hold quite as well as some of the headline figures talk about. And the thing I will leave you with is that nitrogen and carbon, whilst mostly coupled, are certainly not always, as a PhD student of mine has recently demonstrated, much to people's alarm. So with that, I'd like to acknowledge funders in particular, a wall of people who have helped me with this. Most of that data is somebody else's rather than my own. Lynn, deservedly at the top, thank you for your support. And with that, thank you very much.
Our final speaker is someone I have the privilege of working very closely with, and I could easily spend the rest of the evening talking about her. She has been a very generous, kind, and inspiring scientist, leader, manager, and mentor to me in the last 2 years, and to many others in this room for many years. It is my honor to invite Emerita Professor Lynette Abbott to share some reflections with us from her journey of 5 decades in UWA, in the farms, in the soil science, and in all the agriculture sector. Ladies and gentlemen, please extend warm welcome to Emerita.
Hello. This is a bit of a different experience to anything I've ever had before, and I'm really grateful to see you all. Anyway, the other thing I'm going to say is my husband wants to go to music practice, and I'm going to be late. So I'll probably be in trouble, but anyway, you can just wait a little bit longer. So he plays in an orchestra. It's very important. We have to have other things we do in our lives. Thank you. 50 years at UWA. It doesn't feel like 50 years, but anyway, I'm going to run through pretty low-key summary of activities. But firstly, acknowledgement of country, and we've heard it already, but I acknowledge the traditional owners of the land on which we are meeting today. I pay my respects to elders past and present, and I also acknowledge Aboriginal people who may be present here today. In addition, I recognise that my work is relevant across Australia and beyond, and I acknowledge the Aboriginal stewardship of these ancient lands. We are very special in our land here, and I really acknowledge that. I'm starting with thank yous, and to be honest, I don't know how to start with this, and I don't want to leave any of you out because all of you have contributed enormously to what I've done. So I'm not telling a story about myself here. I'm telling a story that I've done with many of you and many people who are not here as well today. But researchers and farmers, very important. PhD students, and the first PhD student, Bill Porter, sitting here. It's wonderful to see. And a lot of these, in fact, nearly all, were also supervised in the early days with Alan Robson. I'll come to that later. But undergraduate students. Now, Siddique actually made the most perceptive comment today. I was a very shy person. I'm not really so shy now. But I was terrified of teaching. And I taught in that lecture theatre over there, in that room. And I had one of the worst experiences of my life in that lecture theatre next door. I'm glad we weren't in that one today. But I learned a lot from teaching. A teacher learns from teaching, and that included all my interactions with farmers. I learnt more than they learned, obviously. Professional staff at UWA has been essential to everyone's wellbeing, and I had particularly wonderful interactions with people who weren't academics in UWA. SPICE, it doesn't exist anymore. It was a blip where we had this fantastic group of people who supported teaching within UWA and resources, and a lot of the resources that I worked on came from interacting with them. The Land Management Society, it doesn't exist anymore, but it's pivotal to what I've done, and I'll mention that later. And also, over the years, we've had funding from all sorts of places, and you probably can't even remember what some of these abbreviations mean, but a lot of people have supported our research, and I'm talking our, it's not me, it's a great team of people over the years, and I appreciate whoever has put money on the table, and I hope that we've done what we can to justify receiving those funds. But I just also here want to say thanks to the people who have organised today, to Rosanna, the wonderful Institute of Ag organiser par excellence, and Hira, and to Siddique and to Matthias for pulling this together. It is 50 years this year since I came, and I really appreciate what you've done to enable this to happen. Thank you very much. And thank you for all coming, so that's great. Now, before UWA, I did have a life before UWA, and I had parents. We were Joneses, And my brother is here. I don't know where he's gone. Oh, there he is. There are 5 of us, and I live on the other side of Australia, and they all live over there. I went a long way away to do what I did. But my parents were teachers, and we lived in the bush. And we lived in a little town called Bingra in northern New South Wales. And it's a lovely town. You should go and visit. It's an agriculture environment, but it doesn't have a big high school. It doesn't have a place where you can go from there to university easily unless you go somewhere else and study. So I went from Bingra to Ingleburn High School, South Sydney, and my journey sounded normal, but Dad moved his school so I could go to a high school where I could go to university. I think something's happened to my slide. I thought I had a slide before that. Can I go backwards? No, must be the next one. I don't know what happened. Anyway, Peter Valder, I'm jumping a bit, I might go backwards in a minute, but Peter Valder was a lecturer in plant pathology at Sydney University. I studied botany, I majored in botany, and I did a project on lichens. So that wasn't meant to be like that. Something's happened funny with the slides. I'll come back to Peter in a minute. But let me say, Dad joined the Soil Conservation Service of New South Wales when I was in Bingra. Now, as a teacher, primary school teacher, he did. You know why? They had a free magazine, and he still had them in his garage when he died, when he was 97. Because they were free, and they were interesting, and they were about soil. And he used to cart us off to soil pits and contour banks and gullies that were being rehabilitated, and we just grew up thinking that was normal. I didn't know I was going to end up in this space. The other thing he had a bit of insight from was that he knew that when we went to this school, Ingleburn High School, for me to do science, I had to learn Latin. But in the school I was in Bingra, they didn't teach Latin. So he arranged, probably surreptitiously, for my French teacher in first year high school at Bingra to get the work from Tamworth High School so I could do it by correspondence, and I did it after school. I learnt Latin after school in first year high school so that when we moved to Ingleburn High School, I could go in the science stream. There'd you be. I don't know. So sorry, it got out of kilter here a bit. So I went to Ingleburn High School and I qualified to go in the science stream because I had done Latin. I wasn't very good at Latin, but I qualified to do it. Then when I went through Sydney University Science, Peter Valder was my honours lecturer and a very inspiring person. It used to be on TV, a lot of stuff talking about plants and all sorts of things. He was wonderful. But I did a project with him on lichens, and it was a symbiosis, soil, terrestrial lichens. A bit like where I ended up, but had no idea that was going to happen. Now, I met Ian Abbott at Sydney University, and he was interested in birds and islands and things like this. And he got a scholarship to go to Monash. That wasn't his first choice. I'll come back to that. But when I went to Monash, I met Lex Parker. That's also relevant because my supervisor at Monash was Lex Parker, who came from here. So I'd met him. Now, getting back to Ian, this is an ad for Ian because he wrote a book on islands of Western Australia, published in 2023. It's a really good book. It's with Andrew Burbidge, islands all the way down from one end to the other of Western Australia, and you can find it on the internet and you can buy it if you like. But it's a synthesis of, and it shows his depth of interest in a wide variety of things, and they just happened to collate this information about lichens. So this was Lex. Something has really gone wrong with these slides. I don't know what. Anyway, they're a bit cockeyed. Now I have to double back here. Ian originally applied to get a scholarship to go to the to North America, and he got a PhD scholarship to do a PhD at McGill University in Montreal. Anyway, he didn't go because he met me. So, so he applied and he got a scholarship and did his PhD at Monash, and that's why I went to Monash. So he didn't go to McGill, but after we both finished our PhDs, he then applied for a postdoctoral fellowship at McGill and worked with the same person, but on a different topic to what he would have done. And he went there to work on the Galapagos Islands, Darwin's finches. Now, that was pretty amazing. So that was, we ended up having a 4-month camping holiday trip. It wasn't a holiday, but there's our little tent on the Galapagos. I don't remember how many islands we visited, but it was quite a number. 2 weeks per island, studying the relationship between the birds and what they were eating, and perhaps why the different size bills occurred across that dimension. It was with Peter Grant, and he and his wife Rosemary they're amazing. Look them up on the internet and you'll see they are amazing. And they've continued to work on those islands, in particular on Daphne Island. And Peter's just written his autobiography, and Rosemary has also written hers, and they're wonderful reads, just for anyone interested in anything, literally. But this was an island, and just for a little aside, I landed on that island, and that required a boat coming up. When the water went up, you jumped off onto the island. There was no beach. I don't know how that happened, but it was terrifying. And we camped on that island for 2 weeks, and Peter subsequently, with his family, went and lived and studied there every year for 40-odd years. It's quite a remarkable story. But the other part, we did go to other islands, and we looked and measured seeds, and you can see our photographic skills were not that good at the time. So, what happened? So Ian did his postdoc. We came back to Australia, and he applied for a postdoc at UWA, which he got. UWA had actual postdocs in those days, and he was awarded the first of them, which was good. So we had a chance to fly or get a train, so we hopped on the train, came across to Perth, and we never went back and lived there ever again, on the other side. So, Department of Soil Science and Plant Nutrition, the building is still there. Lex Parker was here. I'd met him at Monash when I was doing my PhD. I thought, knock, knock on the door, and have you got anything for me to do? And he did. So I did some research with John Watson for a little while, but he had applied for a postdoc to work on mycorrhizas, and I was just in the right place at the right time, which was lucky. You can be lucky. You take advantage of luck. But soil science and plant nutrition in those days was pretty amazing. It was a department. We taught across science and agriculture. But Lex Parker was a microbiologist. Alan Robson came back at that time that year. He'd done his PhD here before, but he came back 1994, just a few months before I turned up. But he then was a lecturer in plant nutrition. Alan Posner, chemist at Graham Amors, or physicist, Bob Jilks, mineralogy, etc. These weren't all professors at the time, but they all became professors, and most of them had personal chairs, which was a different sort of, a pretty high level category in those days. It's pretty unusual to have this concentration of soil scientists, and then me. And Siddique mentioned I was quiet and shy, you can imagine. So, but this is how I ended up there, and I'm still in that building. So the funding was originally from the Australian Wool Corporation, and I had a postdoc for 3 years, and we kept the funding, and we being Alan Robson and I, for 20 years. Past research, mycorrhizas, and Chris, where's Chris? Chris and Martin Sheltimer were funded from those research grants, and I think we just had a ball. It was an exceptional experience that we had. So, Alan, I found a photo in our corridor of the soil science building, and over the last few very sad weeks and days, we've seen photos of Alan as Vice-Chancellor and looking in his suit and very important, and everyone saying the most amazing things about him, which we believe, and they were totally true. But sadly, on Friday, we said goodbye to Alan in the beautiful Winthrop Hall, which he loved, and so I just thought it'd be nice to see a photo of Alan when he came. And I don't know the date of this one. Maybe Nicole. Where's Nicole? I don't know if you know how old that photo, but it was about when he came here. Red hair, that's a black and white photo. So I honestly pay my respects to Alan. I wouldn't have had the career I've had and the opportunities I've had. Not everyone gets to work with a person of his calibre. I just thought all these people were pretty normal, and I ran as fast as I could to keep up with them, but really it was an exceptional group of people, and Alan was the leader of that exception. So, you probably can't see this. That was 1974. We had a head boss in our department who took photos of us each year, which was run, and this man on the left was always lined us up for photos, but I'll show you this one because it's identifying a few people more clearly. You can see me hiding on the corner. And you can see Nancy on the other side. Nancy was a PhD student, and for those of you who don't know, Nancy is now a professor of soil science in our back. We got him back after all these years. Bill Simmons, right at the top, was our amazing chemist, and the plant nutrition group was first class, and you had to do it all properly. Bill was the one who ran that lab. And Kevin, down here, and Kevin is 95, and it was wonderful to see him last week. And Kevin ran the glass houses, and these two people, Bill and Kevin, were amazing in this space. Then Bob Jilts and Alan Posner. And, you know, this was an amazing team. I don't know if you can see yourself there. Some of you will. You can look at it later. But you notice I've highlighted Ian was there. Ian first had his postdoctoral fellowship in zoology, and then he ran out of money, which sometimes happens. And then Lex Parker gave him some temporary money, and he worked on earthworms and soil animals in soil science for a couple of years before later moving to a job that he maintained in the forest department and its subsequent name. So if you want to see the photos, they're in the corridor of soil science, and a little bit of a laugh there. So Alan and I set about to look at what mycorrhizal fungi were, and I set about setting up pot cultures. Now, pot culture might say, what's a pot culture? We picked these spores out of soil, and we put them into soil that had been sterilised, sort of, and grew them up with plants to see what happened. And pot cultures, to me, is a really key thing, because these fungi won't grow. You can't grow them in cultures. They won't grow that way, though some people are finding little ways to do this sort of thing. But we looked at them, we used microscopes, we looked at these fungi under the microscopes, and we set up pot cultures. And I maintained them with people like Chris for many years, and we had a fantastic, and we learnt a lot about these fungi over time. Now, I'm saying this because I think this is fundamental to understanding an organism or a group, and that's my approach, and it's continued. But these fungi get into roots, the roots let them in easily, they have an interaction, a symbiotic interaction with the plant, they exchange nutrients and carbon, but it's just a universal association. Most plants form or let these fungi in, except for lupins. Lupins and canola, they don't like them. But look, it's really fascinating that plants generally let these fungi walk all over them. Why does that happen? So this has been my journey, understanding the ecology, relating it to the plant nutrition. And this is a classic photo where when you have no phosphorus in the soil, putting the fungi in is not going to help you to get phosphorus into the plant. When you've got lots of phosphorus in the soil, the fungi are not going to help you, but it's that intermediate level. And basically these fungi help make more efficient use of phosphorus. Now, that's where we were then, and Alan really was the driver behind the plant nutrition and sorting out a lot of this stuff. But it's about making more efficient use of nutrients, and now we know it's also about efficient use of water, and there's a lot of complexity understood about these things now. We did field trips. This is Badgingarra, and Alan and Kevin there and Bill in a field experiment at Badgingarra. But David Jasper, who wasn't able to come today, but David was a student in ag, and he came back and led the work on mine rehabilitation. And while we say we're trying to look to see whether we could get mycorrhizal communities back into disturbed land, what we learnt from extreme disturbance was relevant in agriculture, in all disturbed environments. So we were trying to understand a fundamental state of these organisms in soil, and it related to agriculture. But I want to also tell you, we had no computers, and we hand-wrote manuscripts, and I did have a typewriter, but we used to cut and paste. Now, on the computers, it says cut and paste. That's where it came from. It was literally cut and paste, sticky tape. So when Alan and I would be writing papers, we'd have handwritten things all over the floor with scissors cutting and sticking things together with sticky tape, literally. So this is where we started, but maybe we did a lot more thinking then. I think, I maybe shouldn't say that, but I think that was a good process. You had to think, and if you had to write it out again if you didn't like it, it was fun. But there are a few things here. Looking back, I think that I was lucky to study in a part of the world which had pretty highly weathered soils, because we asked lots of questions that other people in other parts of the world didn't ask. So that was a benefit to work in this environment, and I'm really grateful for that. But also the challenging Mediterranean hot, dry summer, that drives a lot of, it forces things, it causes little problems for people to deal with the dry climate, but it made us think more. And it's isolated. Now, why would it be useful to be in an isolated part of the world? Because you didn't know, well, perhaps with the computers now, internet, you know, but in those days we didn't know what other people were thinking. We had to wait for new phytologists to come out, probably 6 months later, whatever. But we were here, and David Heyman from Rothamsted came through as a visitor because he'd been invited by the New South Wales Department of Agriculture to tell them about mycorrhizal fungi. Now, that wasn't us starting that. That was, we heard he was coming, we grabbed him on the way to Sydney. And I'm really grateful for that. He taught us basic understanding of mycorrhizas. But after that, we just did it our way. But we did it our way based on someone else's information. And David was a leader in mycorrhiza research at Rothamsted. Now, I'll start to speed up now. I became a lecturer in 1987. I taught over the subsequent years in every unit that was in the university, I think. And sometimes it was because there was no one else to teach it, but other times it was because the units were changing. But one thing that did happen is that we integrated soil biology in the soil science teaching. And in a way, I was doing myself out of a job because there used to be separate units in some of these things, but then they became integrated. And that has persisted. I think UWA has got a lot of integration of soil biology in their soil science teaching. This is a compost. I think this is sea-wise, actually. This is an overhead slide. We used to use overheads, you know what they are? Here's maybe. And so this was one that you can't see the bits of it, but me trying to integrate the whole of the integration between soil and plants and microbes in one slide. That's the whole of what I taught. There's a lot of information there. But I did do some other really interesting things. I was involved in a project with Europe. We exchanged 72 master's students between Europe and Australia, between 4 universities in Australia and 4 universities in Europe. And Felicity was one of those students who went to Denmark and spent 6 months. This was an amazing exercise, and I really am grateful I was able to do that. I also taught in Kerala, thanks to Siddique, and he arranged for me to visit several times to the university there. And we taught I was teaching an organic agriculture class at the time, and it was a lot about soil biology. And then I'm teaching soil biology, but I look out and there wasn't any soil biology outside of what I was doing, because, you know, everything was soil fertility, and soil fertility was chemistry. And so I did wonder what I was doing and what was wrong. Why wasn't what I was doing part of mainstream? And that was true, it wasn't. So up until many years later, if you looked up soil fertility, it was always about, you know, nutrients really. And of course people acknowledge other bits. But so perhaps it was equally around this, and that's why biology should fit equally with the others. But as you get deeper, you realise it's a lot more complicated. Because it's easy to think what the chemical fertility might be, but the trouble is part of that is actually biology, because it's the nutrient cycling, it's the organic matter transformations. So it's not separated from biology. And then the physical fertility is not separated either, because the fungi, the hyphae, the exudates all interact with the physical side of the soil. So here, start with thinking fertility is about chemistry, and then so you think, oh, well, how does the biology fit in? And then you realise it's all tangled up. And so you can't really go back to thinking biological, chemical, physical fertility, because it's just all tangled up. So it's easier to think of one and another, but it doesn't work like that. Now, Dan Murphy and I edited a book on soil biological fertility in 2003, and we got lots of people to talk about components of soil fertility. But from then on, we started using this concept of soil biological fertility, which is quite widespread now. So let's think about biological fertility. In natural ecosystems, no one's out there fertilising the plants to grow. Everything is working with a biological base, and the soil type and the chemistry and the physics. It's all there, but it's going. It's biologically fertile because it's doing its own thing. And that's, I come from a botany space, and this was interesting to me. But this is happening very slowly. It won't grow a wheat plant, especially in our soil. But it's a natural system, it's ticking along. So what about in agricultural systems? You can still have a biological base, and we've heard that today already, but how do you actually get the most out of that biology? It's very easy to override it with putting nutrients on. You can do away with mycorrhizas, you can do away with rhizobium, you can do away with these the obvious biological systems by putting the fertiliser on, don't worry. And that's a risky, or that avoids risk, so it's a bit challenging to decide how to pull back from keeping the biology alive, active, doing its thing. So I don't think it's easy to decide, but theoretically we should be using the organisms in the soil to get the most benefit that we can, and that is the challenge. If you're working with soil biology, it's about understanding complexity. And simple things like, when do you sample soil? You sample it and you might get one answer. You sample it again and you get another answer. So having that flexibility around complexity is essential. A lot of people want to measure and test for soil biology, and you can measure something that's there. You can, how much of an organism or a group is there. You can measure what they're doing. So the same things might be done by different organisms. So groups can do the same thing, but you might measure different amounts of individual groups. But if you measure this, what does it mean? If you measure it in summer, in winter, you might get different answers. So it's not straightforward just going and sampling soil and say, oh, it's good, it's got all this stuff happening in it. And it doesn't really give you a measure of the health of soil or the quality of soil. It's understanding in relation to the soil type that's going to be more important. So as you've heard before already, most of these biological activities in soil are related to the carbon cycle in one way or another. And I keep showing these slides of soil animals running around. You might think, why? Because they reflect that this stuff's alive. That's the point. It's not to say they're the most important thing, but the soil is living, and these organisms reflect the bacteria and the fungal communities. They're feeding on them quite often, on the nematodes. They reflect the things you can't see, and that's why in a lot of my demonstrations I use this. A little bit of history. We started out with, before the fertiliser industry got really going, you know, manures and all sorts of things, right? Then the fertiliser industry, and I came at the time when, you know, that industry was at its peak. The knowledge that was gained in fertiliser use and practice is amazing in Western Australia. You had the microbial inoculant space, where the rhizobium inoculant, which is a gold star inoculant for soils, well, for legumes anyway, absolutely. And a lot of that happened down this, in our soil science building. Also at Sydney University, but this was a hotspot of that early knowledge. And I visited Dave Chattel, who was a PhD student with Lex Parker just the other day. These people were at the forefront internationally of the rhizobium industry developing inoculant. But then you get more integrated pest management type of activities, and then today is something else. Because today people might think, oh, we can make money out of microbes. We can add them to soil and we can make some money. So we've got this interesting situation where, can you purchase soil biology? Can you go and get some and put it in and fix it? And I basically say no, apart from rhizobium, because the reason you haven't got what you want is because the conditions are not right. So it's all about management, get the soil right, but then maybe augmentation with something. And there's a suite of things happening at the moment with testing products. So, but it's an interesting turnaround where biology was forgotten, and now there's a lot of interest, and not just more overseas than here, actually. Farmers. Now, these two people, Caroline was a graduate in agriculture from UWA, and Rob and Caroline have a farm at Wagen, and I learned a lot from them. They do mixed pastures, and Zachariah and I worked on a action on the ground project from the Australian government, and we published a paper with farmers because they were an integral component of understanding the introduction of these pastures and how it affected soil carbon over time. Now, there's another person who's in this room, I think, maybe still, Peter Nixon. He's sitting right in the back row. Peter Nixon is the first farmer who came in my office in the soil science building to ask a question about soil biology, and it just reminded me I didn't know anything about it. Now, I was going to show a video. I'm not going to show it now because of the time, but this is a little animation that's on the Soil Health app, and I'm going to quickly go past it so it doesn't take another 2 minutes. But this is the title, and I'll just let you say that. And this is my last slide, work-life balance. I've done a lot of other things. I've been involved with industry, growing olives. I've been on Questacon committees. I've been all over China and other places. I grow a garden. I've got a family, and I appreciate what they do to put up with all of my antics. But most importantly, I learned transcendental meditation in 1987, and I think that if I hadn't, then I wouldn't be standing here talking to you about all this stuff, because it really was the best thing I ever did. And to finalise, I thank every one of you, because I know I haven't been mentioning all the people who have done all the things, but I do, in my heart, thank every one of you, because without you, I haven't done this. And I'm delighted that some of the students have come from Indonesia today. I just couldn't believe that. That's amazing. Now they're professors. So, and they have wonderful opportunities. I see what they're doing on Facebook, and so they have wonderful interactions with their people. So there's a lot of other things and people who I haven't mentioned, but thank you for coming, and I really am grateful for this day. That's the end.
Thank you everyone for your time and coming over today and hanging around with us for such a little bit of later time than it was expected to close. But thank you very much for coming over. And